round of applause to welcome our esteemed dignitaries and distinguished guests for the inaugural ceremony of National Conference of Enhancing Ethical and Sustainable Animal Research, aligning infrastructure and welfare with the SDGs. We are deeply honored to welcome the chief guest of the event, Dr. S.K. Datta Sir, Joint Commissioner, Animal Welfare and Member Secretary, CESEA. We are equally delighted to welcome the Executive Dean and Registrar, Lovely Professional University, Dr. Monica Gulati ma'am. We also welcome the guest of honor, Professor Dr. Atul Nasa Sir, Pro Vice Chancellor, STD University. Along with him, we are also joined today by our distinguished keynote speakers, Dr. Vijay Bhalla Sir, Dr. Neeraj Khatri Sir, Dr. Vivek Tyagi Sir, Dr. Puneet Kumar Bansal, and fellow esteemed dignitaries and speakers as well. Thank you so much, our respected dignitaries, for joining us this morning. We request you all to kindly take your seats as we prepare to start this significant day.
This National Conference on Enhancing Ethical and Sustainable Animal Research, Aligning Infrastructure and Welfare with Sustainable Development Goals is organized by the School of Pharmaceutical Sciences, Lovely Professional University in association with the Committee for the Control and Supervision of Experiments on Animals, CCSEA, Ministry of Fisheries, Animal Husbandry and Rearing, Government of India, and in collaboration with various scientific and academic institutions. This conference serves as a platform to promote ethical, humane and sustainable practices in animal research, bringing together scholars, policymakers and scientists to share insights, innovations and experiences that help align scientific progress with animal welfare and the sustainable development goals. I would also like to inform our dear audience and esteemed dignitaries that this conference will be streaming live on the official YouTube channel of School of Pharmaceutical Sciences.
Ladies and gentlemen, please give a huge round of applause as we commence this conference. And now, to commence the formal proceedings, we would like to request our Executive Dean and Registrar, Lovely Professional University, Dr. Monica Gulati ma'am, and our Chief Guest for the event, Dr. S.K. Datta Sir, Joint Commissioner, Animal Welfare and Member Secretary, CCSEA, along with Professor Dr. Atul Nasa, Professor Dr. Vijay Bhalla, Dr. Neeraj Khatri, Dr. Vivek Tyagi and Dr. Puneet Kumar Bansal Sir, to kindly join us on stage and take their respective seats. Ladies and gentlemen, please give a huge round of applause as our dignitaries are making their way to the stage.
Ladies and gentlemen, जोरदार तालियां होनी चाहिए. Ladies and gentlemen, as we celebrate the spirit of this remarkable conference, we take this moment to extend our gratitude through a floral welcome ceremony. We begin by requesting the Executive Dean and Registrar, Lovely Professional University, Dr. Monica Gulati ma'am, to kindly present a floral welcome to our Chief Guest of the day, Dr. S.K. Datta Sir, Joint Commissioner, Animal Welfare and Member Secretary, CCSEA, Department of Animal Husbandry and Rearing, Ministry of Fisheries, Animal Husbandry and Rearing, Government of India. Next, we request ma'am to kindly present the floral welcome to Professor Dr. Atul Nasa, Pro Vice Chancellor, SGT University, Gurugram, Haryana, and President, Indian Pharmacy Graduates Association, IPGA.
Ladies and gentlemen, please give a huge round of applause as Sir is being presented with a token of gratitude.
We now request ma'am to extend the next floral welcome to Professor Dr. Vijay Bhalla, Dean ICIA, SGT University, Gurugram, Haryana, and Treasurer, Indian Pharmacy Graduates Association. Thank you so much ma'am. Now we request Dr. Vimlesh Kumar Sir to continue the floral welcome by presenting the next bouquet to Dr. Neeraj Khatri, Chief Scientist, CSIR, Institute of Microbial Technology, Ministry of Science and Technology, Government of India, Chandigarh.
Ladies and gentlemen, let's fill the hall with a huge round of applause.
We may now request Sir to present the next floral welcome to Dr. Vivek Tyagi, Senior Consultant, CCSEA. Ladies and gentlemen, जोरदार तालियां हो जाए सर के लिए. And now we request Dr. Vimlesh sir to present the next floral welcome to Dr. Puneet Kumar Bansal sir, Department of Pharmacology, School of Health Sciences, Central University of Punjab. Each of these tokens is not just a mark of welcome but a symbol of growth, reminding us that every idea, like every flower, needs care and consciousness to flourish. Now I may request Dr. Navneet Khurana sir, Head of Department of Pharmacology, to kindly extend floral welcome to Dr. LT Singla sir, Guru Angad Dev Veterinary and Animal Sciences University, Ludhiana. Zordar taaliyan ho jaye. Next, I request sir to welcome Dr. Amritpal Singh, Professor, Department of Pharmaceutical Sciences, Guru Nanak Dev University, Amritsar. And now, Naveed sir to welcome Dr. Dharmendra Ahuja, Principal Mentor, Experimental Pharmacology Series Software, Bureau for Health and Education Status Upliftment, Rajasthan.
Ladies and gentlemen, जोरदार तालियां हो जाए.
Thank you so much, sir, for presenting this symbol of respect and harmony. Let's have one more round of applause for our distinguished speakers. Now, may I request Dr. Sheetu, Head of Department of Pharmaceuticals, to kindly extend the floral welcome to our distinguished dignitaries. To begin with, we extend our floral welcome to Professor Dr. Alka Biotra, Global Anti-Doping Scientist, ISO WADA Scientist Leaders and former Director WADA Labs, India and Qatar. Please welcome her with a big round of applause, ladies and gentlemen. Next, we extend a warm welcome to Dr. Harish Chandar, Deputy Director, Quality Control, National Institute of Biologicals, Noida. A big round of applause for him. Next, we welcome Professor Dr. Nirmal Singh, Professor and former Head, Department of Pharmaceutical Sciences and Drug Research, Punjab University, Patiala. Let's have a round of applause for him, guys. And now we extend the floral welcome to Professor Dr. Pawan Krishan, Professor and former Head, Department of Pharmaceutical Sciences and Drug Research. Let's have a big round of applause for him. Thank you so much, ma'am, for presenting this beautiful gesture of appreciation, and thank you to all our dignitaries for gracing this occasion and accepting our floral welcome. Ladies and gentlemen, let's celebrate them once again with a big round of applause.
Now, to mark the auspicious beginning of this conference, we now move towards the lamp lighting ceremony, a sacred tradition that symbolizes the victory of knowledge and the spread of wisdom and positivity. Now, to grace this auspicious ceremony, we would like to invite the respected Executive Dean and Registrar, Lovely Professional University, Dr. Monica Gulati ma'am, to kindly come forward for the lamp lighting ceremony. Along with her, we request all our distinguished speakers, Dr. S.K. Datta sir, Professor Dr. Atul Nasa, Dr. Vijay Bhalla sir, Dr. Neeraj Khatri, Dr. Vivek Tyagi, Dr. Puneet Kumar Bansal, Dr. L.D. Singla, Dr. Amritpal Singh, Dr. Dharmendra Ahuja, Professor Dr. Alka Biotri, Dr. Harish Chandra, Professor Dr. Nirmal Singh, Professor Dr. Pawan Krishan, and Dr. Bumlesh Kumar sir to kindly join in for the lamp lighting ceremony. Ladies and gentlemen, please give a huge round of applause for our dignitaries. We also invite Dr. Damanpreet Singh to join us for the lamp lighting ceremony.
दीप प्रज्वलन का यह पवित्र क्षण नए विचारों, नई संभावनाओं और नए संकल्पों की शुरुआत का प्रतीक है. As the lamp glows bright, may it inspire us all to carry the flame of integrity, innovation, and compassion in everything we do.
Dear audience, please give it up for our dignitaries. Thank you so much, everyone. We request all our dignitaries to kindly take their respective seats as we continue with the next segment of the program.
Next, I would like to request Dr. Bimlesh Kumar sir to present a floral welcome to Dr.
Damanpreet Singh sir. Ladies and gentlemen, please give it up for our dignitaries.
जोरदार तालियां हो जाएं, ladies and gentlemen.
Now to officially commence the inaugural session of the National Conference on Enhancing Ethical and Sustainable Animal Research, may I invite Executive Dean and Registrar, Lovely Professional University, Professor Dr. Monica Gulati ma'am, to deliver the welcome address and share her valuable words of encouragement. I expect a huge round of applause as we welcome ma'am.
Good morning. Good morning to all the children and all the delegates. Delegates who are there in the offline and online mode both. So, welcome to all of you. And first of all, I would like to welcome Dr. S.K. Datta. I recently visited India visited New Delhi to invite him, and I could see, how busy he was. Sir, thank you so much for joining us despite your very busy schedule. And the Dr. Tyagi, who has been instrumental in arranging this event. Thank you, sir, for joining us. Now, I would like to welcome Dr. Atul Nasa, who inducted many of us, many of my colleagues, including me, into the very prestigious organization, Indian Pharmacy Graduates Association, more than a decade back. So it was in 2014, from this podium only he had, motivated many of us to join IPGA as an organization, and I think almost all the faculty members of our department or of our school are already the members of IPGA. So thank you, sir. We welcome on behalf of the School of Pharmaceutical Sciences, I welcome you, along with Dr. Bhalla, Dr. Vijay Bhalla, who has been a strong support to us throughout. And then I would also like to welcome Dr. Alka Biotra. We had been after her for almost a month now. She happens to be, the driving force behind the dope testing not only in India but across the world. She has I was talking to her in the morning, she has kind of introduced the concept of officially the dope testing into India, almost single-handedly, she was leading the organization which was created for this only, and she has already given us many meaningful suggestions. Ma'am, we welcome you to this wonderful event which is going to be held now. And then I would also like to welcome, 4 of my colleagues who are stalwarts in pharmacology. The entire country knows about them. Personally speaking, since I am not from pharmacology background, Dr. Nirmal Singh, Dr. Pawan Kishan, who are from Punjabi University, Patiala, and then we have Dr. Amrit Pal with us and Dr. Puneet. So personally speaking also, I have learnt so many things from them. As far as the animal experiments are concerned, I was almost illiterate, being from formulation science. I was not knowing much about it. The kind of grip, the kind of hold they have on the animal testing ethics and animal testing methodology, it is unparalleled. Dr. Neeraj Khatri, who is from IMTech, welcome sir to this event. And many of my students sitting here would recognize him, who have got the training from IMTech time to time. He has been kind enough to train our students at personal level also. The children, Doctor, when children came back, they told me that Doctor used to interact with us at personal level also. Thank you for educating my children. Then Dr. Dharmendra Huja, he has traveled a long distance. Students, let me share with you the software on which you learn the animal experiments before venturing into your M.Pharm. They are all, the brainchild of Dr. Ahuja, and he has been supplying this software to the pharmacy colleges across India, the entire nation, and I would compliment you, sir, for saving the lives of so many of the animals. Dr. Chandra from NIB, I think this is his first visit to us. So welcome, sir, to LPU. And I hope this is not going to be the last visit. We are going to collaborate and keep on working in collaboration with each other. Similarly, Dr. Damanpreet, we would like that, first of all, I welcome you on behalf of my students and my faculty. And then I hope this collaboration goes beyond this visit. Singla sir has been there. So, yeah. So, children, all the know-hows of the animal house, how it is to be prepared, what are the technical parts to be taken care of so that the animals on whom you are going to do the experimental practicals, how is the breeding to be done and everything. Sir, we stand indebted to you. We are obliged to you for, guiding us on every step of creation of this animal house and then the breeding center. Thank you so much for accepting our invitation. Thank you. And children and delegates both, I feel very privileged and proud to share it with you. We have had many conferences earlier also with a lot of fanfare. Like in 2014 also we had a conference of IPGA. But believe me, this is the first time we have such a strong galaxy of intellectuals with us. Everyone has something to give to you. So my request to each one of you is try to learn the maximum. It is very rare that such accomplished people are there on a single forum at a single event. So try to learn the maximum. If you have any questions, please do ask them. And try to make some collaborations. Those children who are feeling sleepy can please have a sip of water and wake up, shake yourselves up, because this is going to be a great learning. That is my promise to each one of you. Thank you so much.
Thank you so much, ma'am, for your insightful and inspiring words. Your vision and leadership truly motivate us to keep serving for innovation with compassion.
Moving ahead to introduce the purpose and vision of this prestigious conference, may I now invite our respected chief guest, Dr. S.K. Datta, Joint Commissioner in Animal Welfare and Member Secretary, CCSEA, Department of Animal Husbandry and Dairying, Government of India, to give an introduction to the conference. Sir, we are honored to have you with today.
Ladies and gentlemen, please give a huge round of applause as we welcome sir.
Thank you very much, Dr. Monica ji, for inviting in such a great conference. I think it is the first time in the Punjab I am visiting, and such kind of conference is happening here on the ethical aspect. And Dr. Atul, Pro-Vice Chancellor, my colleagues, Dr. Neeraj Khatri, and all distinguished guests. If I take the name, that time is very short because you have to learn many things from our distinguished guests, learned scientists, and all. I think you will learn a lot on these animal experiments since you are entering in a new area because nowadays we have to be a very balanced life between animal and human. So I used to give some kind of in this type of seminar, some kind of lecture series within half an hour. So why this animal ethics and animal welfare, it's very important for our life. So I am not going to these rules, regulations and all these things that you will come to know. My colleagues are here. Dr. Tyagi will make aware you on this. So think these children of this new generation, so they should learn each and every objects of our universe and object of our environment, so how we can interact with them. So with this thinking, see, this environment is impacting everybody. Nowadays this situation is such kind of situation that we are living in a very toxic environment. So toxic I am saying this in every sense. So we have to keep our environment in good condition. So similarly, this animal kingdom, it's a part of this environment. They are contributing to our environments, and in fact, they are contributing to our human day-to-day life. So somebody, some person, but many of us think so. It's animal. This life has no value, and what contributions except the eating, and they are providing food, and they are producing food for us, milk, meat, and eggs. So these are food-producing animals. So if you think deeply, that animals, what they are contributing in our life, right from this, I would say that if you wear a shirt and if you wear a sweater, that is a contribution of the animal. But starts with the animal wool first thing, and thereafter we eat. They are providing food, they are providing nutritional supplements, and they are sacrificing their life for the betterment of the human being, for development of various drugs, so on which you are going to work. So until and unless you realize their sacrifice or dedication in the development of human, to safeguard the human life, so you cannot handle them in a proper way. When you are not, why I am saying this word, you see, suppose we are in a stress, somebody wants to do some experiment, suppose in simple context, we are in a stress condition, and we ran across to the dispensary, and we are going for a ECG test. So what result you will get? So everywhere when you are conducting experiment on animal, that animal should be in a very good state, so that the values you are getting from those studies, that will give the good result. So if you are not keeping this animal in a good conditions, so you will not achieve your desired goal, that means your results. So in that case, you will suffer and suffer and suffer. I came across all these situations when we are in the master students, so there is no such kind of this system, animal experimentation, keeping animal in a good conditions, keeping animal in doing experiment in the BSL4, BSL3 facilities, and such kind of conditions. So we used to get the bad results. The results are not corroborative with these results of which are available in the international journal and all these things. So when your results are not coming in a good conditions, your paper will not get published, and you will lost your all efforts. So those people who are interested in conducting such a not only I am not I was telling madam that why you have called this only the pharmacy students, because animal experimentation now it is required everywhere where this biological interventions is going on. So everywhere you get the animal, even IIT they have animal house, and they are studying on the animals. Where everywhere we have 1700 odd institutions registered with us. Everywhere some kind of animal experimentation is going on. So without animal experimentation, without handling the biological life, so you cannot go ahead in the biological science. So biology defense, this physiology differs in the many conditions to which you cannot simulate in a different alternative methods. So animal experimentation is important. And you students, when you enter into the master's degree, because in undergraduate animal experimentation, it is not that much of requirement. Only you can learn this handling, how these animals are being handled. So when you are will enter into the master's, so you will every time you require to handle the animal. So our intention of conducting such kind of seminar and workshop is to make aware about these ethical conditions of the animals, and rules and regulations are in place in our country. You can't believe India is the oldest country. First in 1890 we have Prevention of Cruelty to Animal Act, that is the British Area Act. In 1819, it's 90 itself, this animal, this act came into force in Parliament at that point of time. Thereafter, 1960, that act was, this 1890 Act was repealed, and this whatever act we are having, that act came into force. So it is a very extensive act. Wherever these animals are abused or animals are there is a chance of getting unnecessary pain and sufferings to the animals. Everywhere it is covered. The act covers everything. Now you see this recently, this 2023, this Bharatiya Nyaya Sanhita, that is called the BNS Act. There also a section, 2 section has been introduced, that is dealing with this animal. So if you are mutilating, if you are killing, you are torturing on the animals, there is a 5 years imprisonment provision is there, and with a heavy penalty. So government, we are also the various rules and regulations, various species involvements, various species, how these animals are being kept, and how what would be the actual ambient condition of these animals in the laboratory conditions, how animals are behave in the laboratory conditions, how we will inject this drug into the animal body, and how the rats are being handled. Everything you will learn to came to know. Without that, please, you are the students, and you obviously publish the paper, and without the animal experimentation and certificate that is called the Institutional Animal Ethics Committee clearances, you cannot publish any paper. So, where our efforts is at that to reach out to these students who are actually handling the animal or will handle the animals, to come and to know these what are the rules and regulations and what are the protocols and the standard operating procedures for handling such kind of animals. So this is the main intention of our this kind of seminar workshop, and our organizations, though we are facilitating animal experimentation. So earlier there was used to that there are some issues with clearances and all these things. So nowadays we have very much streamlined with this all these experiment. It doesn't mean that every time you will abuse the law and you will violate and you will flout all the norms of the CCCA. In that case, we are also taking very serious actions on these violators. We promote the, we cannot say at this time promote the animal. We are also working on these alternatives of the animal experimentations. We have some collecting informations and repositories which are the animal experimentations. And my suggestion on the these instructors and the PIs who are developing protocol. So animal ethics works in 3 R's. So 3 R's means these animals are sentient beings, they have a pain. So when these animals are sentient beings, try to restrict this experiment in a non-sentient animals, that is called these animals feeling less pains. Whenever you are you conduct a study on these higher up animals, so you have to take care how to rationalize this use of the animal. So try to reduce the number of animals, and how to reduce the number of animals, that is the art of framing the research work. So if we require 80 animals, and how we can these 80 animals can be reduced if it is possible. The statistical data is sufficient enough below the 80 animals, you do it, or vice versa. If you require more animals, you can design your study accordingly, so that the repetition of the animal experimentation should not happen. Second thing is that you try to, this instructor should also try to make aware about the students how to reuse the animal and how to rehabilitate. What are the principles, these animals are there. So everything you will learn and you will follow the CCCA guideline. So animal experiment, I always emphasis on this repetition of the experiment and repetition of the use of animals. Now this internationally, many countries, specifically the European Union, they are downsizing the animal experimentation. I think they have a target of waiving of all animal experimentation by 2030. So now this animal experimentation will be the little bit of these conditions, because many organizations are working against this animal experimentation. And so we'll face a we are facing difficult situation, will be facing difficult situation when European countries will stop the animal experimentation. Second, another issue is that this India is flourishing on the pharma sector. So pharmacological sectors and this we are a big CROs working here. So huge earnings are coming through the contract research. So where the animals, large number of animals are being used, and issue of controlling the issue of the ethical treatment of animal is also a major concern of the CPCCA. For that we are always vigilant on this, on their working. So all these rules and regulations and systems are in place. All these rules regulations are getting updated day by day, and everything is available. We have a very good website. Everything is online. You can submit the application through online, whatever you required, and we are clearing this very expeditiously within a month time, unless some administrative problem is there. So within a month's time, we are clearing all such kind of applications, and whatever requirement you require, we are always here to answer and reply through email correspondence, through all these things. We are here. But we are also at the same time, we are very strict on taking action nowadays, because every informations are gathered in the central level. Don't think that we are sitting in Delhi, we are not getting any information. We are getting every information. So we don't expect that those who are family of the CPCCA, their complaints should come from the third party. So don't expect on that, because we are working very hard to streamline our systems, animal experimentation, so that no institution can face any difficulties for getting clearance from the CPCCA within the framework or guideline. So whatever problem you are facing, always try to contact with us, and our nominees try to take work from the nominees, and we don't expect that third party communication from anybody complaints, any institutions complaints. So in that case, we are very stringent. We take actions against anybody. We do not leave anyone on this nowadays, because we are not now, this body has totally changed in their working. We are facilitating. When we are facilitating, we are strict as well. So with this, I think this seminar, these students will learn and the animal ethics that I took a longer time. So my suggestion would be, these students don't feel sleepy, because if our animals are good, in good health, we will all be in good health. So this is not my saying. If you search out this international media and all, so nowadays this is 65 to 75% of the diseases are coming from the animal. That's all genetic borne disease, called the genetic diseases. So why we are saying so, and international communities are little worried, and we are working in a common platform. It is called the One Health platform. We are working. So how to stop this spillover of the disease from animal to human, human to animal. So until and unless animals are in a good health, we will not get the good quality food, we will not get the good quality environment, because everywhere the disease will spread and people will suffer. So animal welfare now is a big issue in the international forum also, and this, we have a World Organization for Animal Health like the WHO. So they are coming up with a big standards, animal welfare standards, and they are really concerned about that. They are really concerned about that, and they are watching every country how they are doing the ethical treatment. And in our country, we are the one of the leading country on animal ethics work. And like that, no country is such kind of guidelines. I think US, they are trying to evolve their own guidelines. So we are very good system, and we are compassionate. In Indian history, we are compassionate. We cannot kill animal like that. So people are, you are seeing the social media and all these things, everything will come immediately on the social media. So animal ethics, you learn the animal ethics and do experimentation with your compassion, and you will get the very good results with this. I congratulate the organizers for working, arranging such a wonderful seminar with these huge gatherings. And thank you very much, and thank you all.
Thank you so much, sir, for providing such deep insights into the objectives of this conference. And now, to set the context for the day and give us a broader academic perspective, we are honored to have with us a distinguished leader with over 4 decades of experience in the fields of pharmacy, regulation, and academics, Professor Dr. Atul Nasa, Vice Chancellor of SGT University, Gurugram, and President of the Indian Pharmacy Graduates Association. Sir, we request you to kindly share your thoughts and set the context for this conference. जोरदार तालियां हो जाएं सर के लिए.
Good morning. On the dais, Dr. S.K. Datta, the Member Secretary of CCSEA and the Chief Guest of today's event. Very dynamic and energetic, I can say, Dr. Monica Golati, the Executive Dean and Research of lovely professional university. My friend from SGT University, Dr. Vijay Bhalla, and Dr. Neeraj, Dr. Vivek Tyagi, Dr. Puneet, Dr. Bimlesh, and I am seeing in the audience Professor Alka, Dr. Amrit Palji from GNDU, Dr. Dharmendra, Dr. Harish from NIB, Dr. Nirmal Singh, Dr. Pavan, Dr. Damanpreet, and all respected audience, and not but last the least, very dear students from pharmacy. सबसे पहले तो मैं डॉक्टर मोनिका का एंड डॉक्टर बिमलेश का थैंक्स करूंगा. इतना प्रेशर किया कि आप सर LPU आइए एक बार. दिस इज़ माय सेकंड विजिट एंड मैडम हैज़ टोल्ड वी केम हियर फॉर वन ऑफ द एनुअल कॉन्फ्रेंस ऑफ IPGA in 2014 and still I remember at that time Shri P.A. Sangmaji, who was the Lok Sabha speaker, he was on the dais along with me and he was the chief guest. So thank you very much Dr. Monica and Dr. Bimlesh for inviting us to this LPU campus, a beautiful campus I can say. I will talk little bit different from animal research क्योंकि मैं देख रहा हूँ यहाँ pharmacy students हैं and my journey of 40 years, I can say more than 40 years in pharmacy profession, from pharma industry to academics and 30 years in regulations. It was a tough journey, it was a challengeable journey, but I always say it was a successful journey because I always say I am a pharmacist. मुझे याद आ रहा है आज से लॉन्ग बैक जब किसी ने मेरे को बोला फार्मेसी का कोर्स करिए, मुझे तो फार्मेसी का ABCD मालूम नहीं था. डॉक्टर मोनिका से भी बात हो रही है, डॉक्टर मोनिका की सारी फैमिली डॉक्टर्स की है, तो मुझे तो फार्मेसी का ABCD मालूम नहीं है. 1977 लॉन्ग बैक 1977 जब इंडिया में 10+2 स्कीम स्टार्ट हुआ फर्स्ट टाइम. अर्लियर इट वाज 8+3 हायर सेकेंडरी. तो गवर्नमेंट ने अनाउंस किया कि आफ्टर पासिंग ऑफ योर मेट्रिकुलेशन डेट इज 10th क्लास आप डिप्लोमा में एडमिशन ले सकते हैं, पॉलिटेक्निक्स में, इंजीनियरिंग में, फार्मेसी में एंड सो ऑन. अच्छा, हमने तो सोचा कि हम 11th एंड 12th करेंगे, डॉक्टर बनेंगे, पर जब गवर्नमेंट ने ये अनाउंस किया कि आप 10th के बाद, मेट्रिकुलेशन के बाद फार्मेसी में एडमिशन ले सकते हैं, तो मुझे किसी ने बोला यार फार्मेसी कर लो. अब हमें तो ABCD मालूम नहीं फार्मेसी की. And I took my admission at the age of 15 years in Diploma in Pharmacy course in Delhi College and completed my pharmacy course at the age of 17 years. अब 17 साल की उम्र जहाँ मुझे पास करना था 12th क्लास, हमने डिप्लोमा इन फार्मेसी किया विद ए बिग क्वेश्चन मार्क. अब आगे क्या करें? आई वेंट टू एम्प्लॉयमेंट एक्सचेंज फॉर रजिस्ट्रेशन, भी चलो जॉब के लिए नाम लिखवाते हैं, बोले मिनिमम एज शुड बी 18 ईयर्स. आई वेंट टू दिल्ली फार्मेसी काउंसिल फॉर रजिस्ट्रेशन, बोले मिनिमम एज इज 18 ईयर्स. आई अप्लाइड फॉर बैचलर ऑफ फार्मेसी कोर्स लेटरल एंट्री फॉर बी फार्म सेकेंड ईयर एडमिशन, बोले मिनिमम एज इज 18 ईयर्स. अब एक साल इतना बड़ा गैप, करें क्या? यंग एज ना हम केमिस्ट शॉप खोल सकते हैं क्योंकि रजिस्ट्रेशन इन, और जैसे ही 18 साल की एज हुई, अडल्ट हुए, तो बैचलर ऑफ फार्मेसी में एडमिशन लिया, फिर पीछे मुड़ के नहीं देखा. एंड दैट जर्नी कंप्लीटेड माय बैचलर्स इन 1983, मास्टर्स इन 1986, क्लियर्ड माय यूपीएससी इन 92 एंड 30 years इन रेगुलेशन. एक ऐसी जर्नी थी कि आई ऑलवेज सैल्यूट टू फार्मेसी प्रोफेशन. क्योंकि यहाँ सारे स्टूडेंट्स फार्मेसी के बैठे हैं. बहुत बड़ा क्वेश्चन मार्क आता है कि वेदर वी हैव टेकन ए राइट डिसीजन और ए रॉन्ग डिसीजन. बट आई टेल यू, you have taken a right decision by choosing pharmacy as a career. Lot of scope, हमारे टाइम में तो scope बहुत limited था. Today, any student who is completing his pharmacy course, whether it is Bachelor of Pharmacy or Master of Pharmacy or PharmD program or Doctorate in Pharmacy, ample scope, I tell you. Pharmaceutical industry is open, R&D sections are there, quality control departments are there, quality assurance departments are there, medical devices, upcoming field. Lot of scope, lot of demand for pharmacy graduates in medical devices. On this, today is 1st of November, Government of India is going to conduct by UPSC the examination for medical device drug inspectors. Coming forth, आज 1 तारीख है. 27 posts or 26 posts of medical device officers are being recruited by CDSCO, diagnostics, CROs, hospital pharmacy, community pharmacy, clinical pharmacy, regulations, and so on. Choice is yours. What you want to do. But simply I will advise you, never stop your career after graduation program, that is Bachelor of Pharmacy. You must go for higher studies. Higher studies means it can be Master of Pharmaceutical Sciences, combination of B.Pharm and MBA, much demanding. Combination of Bachelor of Pharmacy and Law, much demanding. Choice is yours. But at least after Bachelor of Pharmacy course, my sincere advice to all pharmacists sitting in this auditorium, you must go for higher studies. Complete your education, higher education. And once you complete your higher education, definitely your goal will be achieved and you will be on the top of your positions. Why I salute pharmacists? I have seen the time, toughest time of my life, COVID, pandemic. And at that time, 2021, when the entire world was suffering from pandemic, I was heading Drug Control Department Delhi. Such a challenge. When this first wave came, if you remember, it was an advisory from government that hydroxychloroquine tablet, HCQS, can be used for the prophylactic treatment of COVID. But it was for medical doctors and who are treating the patient. और हुआ क्या? Everybody started, sir, हमें hydroxychloroquine चाहिए. My friends, my relatives, my circle, sir, we want HCQS tablet, and people starting this medicine as a self-medication to protect themselves from the first wave of COVID, but it was not required at all. At that time, the President of USA requested Modi ji, हमें hydroxychloroquine चाहिए, HCQS. direction from PMO office that as much as hydroxychloroquine we can manufacture, only one company used to manufacture SCQS, that was IPCA Laboratories, IPCA. But production बड़ी, हमने पूरी इंडिया को हमने एक्सपोर्ट किया, and proudly I say India is the pharmacy of the world nowadays. Second wave आया, और आपको याद होगा second wave में lot of people casualties due to demand of oxygen, due to the demand of life-saving drugs, due to the hospitalizations, blood bank issues. But entire country was under lockdown, but it was the pharmacy profession. It was the pharmacists working in pharmaceutical industry. Pharmaceutical industry was not closed. And all the pharmacists working in R&D section, quality control department, quality assurance department, manufacturing department, hospital pharmacy, community pharmacy, clinical pharmacy, they were working day and night to save the entire country. And that's why I salute to pharmacists, and pharmacists are the COVID warriors. This is the pharmacy profession. मैडम ने बोला हमने यहाँ पे एक बहुत बड़ा कॉन्फ्रेंस किया IPGA का, इंडियन फार्मेसी ग्रेजुएट एसोसिएशन का, आज से 10-12 साल पहले, जिसका मैं नेशनल लेवल पे प्रेसिडेंट भी हूँ. We always try to host all these conferences in the entire part of the country and inviting all the young pharmacists to participate in the conference. And why? What is the advantage of attending a conference? What is the advantage of participating in a conference? I will tell you. Today, you are meeting such a galaxy of scientists, speakers, resource persons from renowned fields. Today, you came to know who is Dr. Datta, who is Dr. NASA, and so on. And this is called networking. And networking का सबसे बड़ा example मैं आपको आज share करता हूँ. 2009, Indian Pharmaceutical Congress was held at Nirma University. 2009 IPC was held at Nirma University, and at that time the chief guest was Honorable Prime Minister of India, Mr. Modi ji. He was the Chief Minister of Gujarat at that time, 2009. We were sharing the dais together, like Dr. Datta is sitting, I was next sitting to Modi ji. तो मोदी जी ने मुझसे क्वेश्चन पूछा 2009 में, नासा साहब इतना बड़ा 10,000 डेलीगेट्स, आपका कॉन्फ्रेंस का मिशन क्या है? 2009. मैं कहा सर, इंडिया शुड बी फार्मेसी ऑफ द वर्ल्ड, आई टोल्ड इन 2009. एंड मोदी जी ने मेरे इस सेंटेंस को पकड़ के, उनकी तो क्वालिटी है, ही कैन स्पीक एनीथिंग, हमसे ज़्यादा बढ़िया बोला, लगने लगा कि वही फार्मासिस्ट है. ही स्पोक फॉर 40 minutes ऑन वन सेंटेंस, हाउ इंडिया कैन बी फार्मेसी ऑफ द वर्ल्ड. हमने उनके पॉइंट्स भी नोट किए कि व्हाट शुड बी आवर फ्यूचर प्लान. 2014 when he became the Prime Minister of India, I wrote a letter to PMO office, sir, heartiest congratulations for becoming the Prime Minister of India. उस टाइम की कुछ फोटोग्राफ्स थी हमारी डाइस की, उन्होंने मेरे उसमें मैंने सेव किया हुआ था मोबाइल में, तो उनको मैंने वो फोटो भी अटैच करके भेजा. मैंने कहा सर, जस्ट फॉर योर रिमेम्बरेंस, we shared the dash during the 2009 IPC at Ahmedabad, and if possible I want to meet you in person. I got a letter from PMO office, 5-minute slot is given to me, you to meet Honorable Prime Minister of India. और जब मैं उनसे मिलने गया at race course at his residence, उनका वही क्वेश्चन था, नासा जी 2009 and 2014 where we stand, whether we are the pharmacy of the world or not. मेमोरी देखिए आप. मैं कहा yes sir, we are developing and definitely will be the pharmacy of the world. And third time once we interacted during COVID time, virtual meeting, जब कोवैक्सीन कोविशील्ड का चैलेंज था हमारी कंट्री के सामने, it was the office of PMO along with the ICMR, WHO Geneva and Drug Control Department, and I was attending that meeting, virtual meeting. मोदी जी बोले, नासा जी 2021, we don't have any vaccine. Remdesivir, tocilizumab, remember at that time which were used for the COVID treatment, they were not meant for COVID. अगर आप गूगल को सर्च करिए, They are used in the treatment of rheumatoid arthritis where the immunity was down to the patients. So it was a trial method, I can say, used by the medical doctors, क्योंकि COVID patients में immunity down था, तो उसी इंजेक्शन को यूज किया जा रहा था. 6 इंजेक्शन रेमडेसिविर के डिमांड में थे. But yes, directions, hard work of drug control department from pharma industry, Bharat Biotech, Dr. Krishanella, Poonawalla from Serum Institute of India, they got approval within 8 months' time. That was a fast track of approval given to go ahead with the Covaxin and Covishield, and we saved the entire world by getting these 2 vaccines. This is the pharmacy profession. This is, I respect a pharmacy profession, क्योंकि मेरे को pharmacy profession की जर्नी ने, डॉक्टर मोनिका से मॉर्निंग में बात हो रही थी, मैं डॉक्टर मोनिका ने बोला मैं भी थोड़ा वो थी, बट विद इन 7 days और 10 days मेरे को फार्मेसी प्रोफेशन के लिए इतना अट्रैक्शन हो गया कि इससे बढ़िया कोई प्रोफेशन है ही नहीं. और जब हम आपसे ये बात शेयर करते हैं, जब मैंने ऐसी यंग स्टूडेंट सिटिंग इन दी ऑडियंस, हमारी ड्यूटी बनती है टू गाइड यू एंड टेल यू ईच एंड एवरीवन कि इट इज़ ए राइट डिसीजन बाय चूजिंग फार्मेसी एज ए कैरियर. लाइफस्टाइल डिजीज, एक एग्जांपल, WHO has given an alarming data. Young generation is suffering from lifestyle disease. आज यंग बच्चा डायबिटी का पेशेंट है, हाइपरटेंशन से सफर कर रहा है, कैंसर इज ऑन राइज. ऑल दीज आर लाइफस्टाइल. और हमारा लाइफस्टाइल है क्या? हमारा लाइफस्टाइल क्या है मैं बताता हूं. एक दिन में 24 घंटे हैं, 24 आवर्स. 24 आवर्स. अगर हम इसको 3 से डिवाइड करें, 8 + 8 + 8, 8 + 8 + 8, सबसे पहले 8 घंटे जो है साउंड स्लीप. वो मुझे लगता नहीं कि आज के बच्चों को आती होगी, क्योंकि रात को 2 बजे तो हम मोबाइल को देखते हैं. सबसे बड़ी एडिक्शन यही है. आई एग्री. Sometimes social media, yeah, they are useful. I agreed. We get lot of information. Artificial intelligence का time है. So many information, chat GPT and everything we used to see on mobile. But 7 to 8 hours of your sound sleep is very important. हम रात को सोते हैं 1 बजे, जब सुबह 6 बजे या 5 जब भी उठते हैं ना, सबसे पहले मोबाइल को देखते हैं, कोई पता नहीं क्या इन्फॉर्मेशन मिस हो गई. ऐसी कौन सी इमरजेंसी है आंख खोलने के बाद ही मोबाइल देखते हैं हम सबसे पहले? नो. सर बोल रहे थे हेल्थ, एनिमल हेल्थ के ऊपर बात कर रहे थे. अगर हेल्दी एनिमल्स होंगे डेफिनेटली हमारी हेल्थ भी अच्छी होगी. और वो हेल्थ अच्छी होगी कैसे? जब हेल्थ इज वेल्थ. और तो 8 घंटे का वो जो रूटीन है, अगर आपकी साउंड स्लीप है, यू आर फिजिकली एंड मेंटली फिट टू स्टार्ट योर नेक्स्ट डे. और नेक्स्ट 8 hours ऑफ योर कॉलेज लाइफ, ऑफिस लाइफ, जहां आप जॉब करते हैं या जहां आप पढ़ते हैं, वो 8 घंटे बहुत कंस्ट्रक्टिव और पॉजिटिव होने चाहिए. पर होता क्या, आई टेल यू, वो 8 घंटे पॉलिटिक्स में जाते हैं. उसकी बुराई, उसका क्रिटिसिज्म. ये एक ह्यूमन नेचर है जिसको चेंज करना है हमको. वो 8 घंटे जो हम पूरी लाइफ के डेली अपने एक ऑफिस में या अपने इंस्टीट्यूट में या अपने कॉलेज में बिताते हैं, वो इतने बढ़िया होने चाहिए, लगे हाँ आज एक कंस्ट्रक्टिव डे रहा. क्योंकि अगर आपका 8 घंटे का वो टाइम कंस्ट्रक्टिव नहीं है, जब आप रात को सोएंगे आप डिस्टर्ब सोएंगे कि आज मेरा इससे झगड़ा हुआ, आज मैंने इसको गाली दी, इसको मैंने क्रिटिसाइज किया, और रात को नींद तो आएगी नहीं, वो नेगेटिविटी रहेगी दिमाग में. एंड डेफिनेटली 8 hours फॉर योर सेल्फ, फॉर योर फैमिली एंड फ्रेंड्स. जो 8 घंटे बचे हैं, आप उसको यूटिलाइज करिए विद ए फ्रेंड सर्कल, फ्रेंड्स, फैमिली. उसको एंजॉय करिए. सम फिजिकल एक्सरसाइज. कितने बच्चे यहाँ बैठे हैं ऑडियंस में जो फिजिकल एक्टिविटी करते हैं? मॉर्निंग वॉक, मेडिटेशन, योगा, जिम, वट एवर इट मे बी. मैं सर से शेयर कर रहा था, डॉक्टर के अग्रवाल मॉर्निंग में हम डिस्कस कर रहे थे, हु वॉज ए लीडिंग कार्डियोलॉजिस्ट इन दिल्ली, जिन्होंने हार्ट केयर फाउंडेशन ऑफ इंडिया को स्टार्ट किया दिल्ली में. लीडिंग कार्डियोलॉजिस्ट, कोई उनको प्रॉब्लम नहीं थी। एंड ही यूज्ड टू शेयर ऑन यूट्यूब वेरियस मोटिवेशनल वीडियोस ड्यूरिंग दिस कोविड टाइम, ताकि लोग मोटिवेटेड रहें, डिप्रेस ना रहें। बट आई टेल यू, जब वो कोविड के टाइम में उनको भी कोविड हुआ एंड ही वॉज नो मोर। सच ए सैड सिचुएशन। Leading cardiologists who used to motivate each and everyone during COVID time without any lifestyle disease expired during the COVID time. और उनकी एक कॉन्फ्रेंस मैंने अटेंड किया, फार्माकोलॉजी का कॉन्फ्रेंस था हार्ट केयर फाउंडेशन की तरफ से। उन्होंने बोला कि अगर आपको पूरे दिन में 30 मिनट्स का टाइम नहीं है फिजिकल एक्सरसाइज करने के लिए, 30 मिनट्स, एक काम तो आप कर सकते हैं, जिस बिल्डिंग में आप रहते हैं, जिस बिल्डिंग में आप जॉब करते हैं या जिस बिल्डिंग में आप पढ़ते हैं, उसमें कभी लिफ्ट मत यूज करिए। गो बाय स्टेयर्स, कम बाय स्टेयर्स। गो बाय स्टेयर्स, कम बाय स्टेयर्स, दैट इज मोर देन एक्सरसाइज। और वो दिन था मेरा दिल्ली सचिवालय में जो ऑफिस था हमारा हेल्थ डिपार्टमेंट का, दैट वाज ऑन 9th फ्लोर। और दिमाग में तो घुसा कि डॉक्टर के अग्रवाल ने बोला लिफ्ट मत यूज करिए, पर अब 9 मंजिल चढ़ेगा कौन। बट आई ट्राइड माई लेवल बेस्ट, और उस दिन के बाद मैंने कभी लिफ्ट यूज किया ही नहीं। why I am sharing this? Again I am repeating, health is wealth। आप बिल्कुल सब समझ लीजिए, आप जितने बच्चे यहाँ बैठे हैं, जितना मर्जी पैसा हम कमा लें, जितनी मर्जी टॉप पोजीशन पे चले जाएं, अगर हम फिजिकली या मेंटली या हेल्दी नहीं हैं, सब कुछ बेकार है, सब कुछ बेकार है। और यही फार्मासिस्ट का रोल है, यही फार्मासिस्ट का रोल है। और रिस्पेक्ट, थोड़ा मैं जनरलाइज बात ज़्यादा करता हूँ क्योंकि बच्चे यहाँ बैठे हैं। व्हाट इज डेफिनेशन ऑफ रिस्पेक्ट? आज की डेट में अगर आप किसी से रिस्पेक्टफुली बात करेंगे, ऑटोमेटिकली आपको रिस्पेक्ट मिलेगी। इट इज रेसिप्रोकल। आज आप एक छोटे से बच्चे को भी तुम नहीं बोल सकते। वो भी एक ऐसी जनरेशन है, हर चीज बच्चा समझता है, जैसा आप उससे बात करेंगे वैसा वो आपसे बात करेगा। और उसका एक एग्जांपल मैं आपको बताता हूँ, रिस्पेक्ट का बहुत बड़ा एग्जांपल, बड़ा टफ एग्जांपल था मेरे लिए। I was teaching in Dipsru University, that is called Delhi College of Pharmacy. Now it is named as Department of Pharmaceutical Sciences and Research. तो somebody told me, यार आप टीचिंग छोड़ के ड्रग कंट्रोल में जाइए। हमने बोला हम तो टीचर हैं, एकेडमिक्स से बिलोंग करते हैं। I applied to UPSC 1991 for the post of drug inspector। हमारा सिलेक्शन थ्रू UPSC था दिल्ली का, सेंट्रल ड्रग कंट्रोल एंड दिल्ली स्टेट का सिलेक्शन इज थ्रू UPSC। रिटर्न क्लियर हो गया। 75 कैंडिडेट दे शॉर्टलिस्टेड, 4 वैकेंसीज वर देयर एंड माय इंटरव्यू वाज ऑन लास्ट डे। 15 कैंडिडेट्स पर डे दे कॉल्ड फॉर इंटरव्यू। अब हम जब 5वें दिन इंटरव्यू देने गए, ये माइंड सेट था यार सिलेक्शन तो 4 पोस्ट का है, अब तक तो हो गया होगा। पर ये भी सोचा कोई नहीं, नहीं होगा, पढ़ा तो रही हैं, टीचर तो नहीं। When I appeared in the UPSC interview with 3 subject expert, 40 minutes grilling, वो जो एड्रेनलिन लेवल अप एंड डाउन चल रहा था, तो मिस्टर चतवाल हुआ जी चेयरमैन ऑफ UPSC, बोले मिस्टर नासा यू कैन गो। तो आई वॉज रिलैक्स, माई एड्रेनलिन लेवल केम डाउन। चलो इंटरव्यू खत्म हुआ। जैसे ही मैं डोर पे पहुंचा तो उन्होंने बोला मिस्टर नासा एक बार वापस आइए आप। तो मैंने बोला व्हाट नेक्स्ट। हमारे में फिर विचार आया यार इंटरव्यू तो बहुत बढ़िया हो गया, अब इनके दिमाग में कौन सा क्वेश्चन ऐसा आ गया जो लास्ट मोमेंट में मेरे को दोबारा बुलाया। कह लगे एक क्वेश्चन पूछना हम आपसे भूल गए, यू आर होल्डिंग ए वेरी रिस्पेक्टेबल पोस्ट ऑफ टीचिंग। टीचिंग इज ए नोबल प्रोफेशन, ऑलवेज टीचर्स आर रिस्पेक्टेड, एंड यू वांट टू लीव टीचिंग जॉब एंड यू वांट टू बिकम ए ड्रग इंस्पेक्टर। एंड इंस्पेक्टर मीन्स करप्शन, वर्ड है ना इंस्पेक्टर। एंड दिस इज नॉट ए रिस्पेक्टेबल पोस्ट इन द आइज ऑफ पब्लिक। परसेप्शन इज नॉट गुड, क्योंकि जिसके पीछे वर्ड इंस्पेक्टर लगाया हर आदमी उसको एक ही आंख से देखता है, ये तो चोर होगा। कई स्टेट ने ड्रग इंस्पेक्टर की टाइटल को चेंज कर दिया। इन हरियाणा इट इज ड्रग कंट्रोल ऑफिसर्स, नॉट ड्रग इंस्पेक्टर्स। In Rajasthan, they have changed the nomenclature from drug inspector to drug control officer because it is a gazetted post. It is a gazetted post. तो उन्होंने जब मेरे से क्वेश्चन पूछा कि नासा साहब टीचिंग छोड़ के ड्रग इंस्पेक्टर विच इज नॉट ए रिस्पेक्टेड पोस्ट, गिव वन रीजन व्हाई यू वांट टू लीव टीचिंग एंड यू वांट टू ज्वाइन ड्रग कंट्रोल डिपार्टमेंट. इट वाज द प्रेजेंस ऑफ माइंड एंड गॉड वाज देयर. मैं सर रिस्पेक्ट इज ऑलवेज कमांडिंग, इट इज नॉट डिमांडिंग वेयर एवर यू वर्क. कमांड द रिस्पेक्ट. बोले बड़ा डिप्लोमेटिक आंसर. मैं कहा सर नो, दिस इज द रियलिटी. कहीं भी आप जॉब करिए, रिस्पेक्ट को कमांड करिए. और जब मैं बाहर निकला एक इंट्यूशन थोड़ा सा हुआ कि शायद सिलेक्शन हो गया, शायद. एंड व्हेन द रिजल्ट केम आई वाज द यूपीएससी टॉपर. व्हाई आई एम शेयरिंग विद ऑल ऑफ यू? क्योंकि आपकी जर्नी, बच्चों जितने भी स्टूडेंट्स यहाँ बैठे हैं, आज बिगनिंग है और आपका फ्यूचर इतना ब्राइट होना चाहिए कंडीशनली कि जो पॉइंट्स मैं आज आप सबको शेयर कर रहा हूं उसमें से एक पॉइंट भी अगर आप अडॉप्ट कर लेंगे वो आपकी लाइफ के लिए बहुत बड़ा सक्सेस का वो बेस बनेगा. बिकॉज आई एम शेयरिंग माई सक्सेस जर्नी. जो जर्नी मैंने चैलेंज भी देखे, फेसिस भी देखे, सिचुएशन भी देखी, पर टॉप पे पहुंचते गए. और लास्ट कंक्लूड करता हूं, हायर एजुकेशन जो मैंने बताया. 2016 मुझे किसी ने बोला पीएचडी कर लो. मैं सर हम तो 54 साल के हैं, अब आई एम 62 बॉर्न. अब इस एज में कौन पीएचडी करेगा? और हम तो ड्रग कंट्रोल से हैं, हमें क्या पीएचडी का बेनिफिट है? बोले नहीं करो. तो आई अप्लाइड फॉर पीएचडी एडमिशन टू एमईटी यूनिवर्सिटी नोएडा. हमने कभी जिंदगी में ऑनलाइन एग्जाम ही नहीं दिया, हमारे टाइम में होते नहीं थे. तो आई वेंट टू एमईटी यूनिवर्सिटी. एक बहुत बड़ा हॉल, 1000 कंप्यूटर लगे हुए हैं एंड ऑल स्टूडेंट फ्रॉम डिफरेंट फील्ड एंड स्पेशलाइजेशन जिनको पीएचडी करनी है सभी कंप्यूटर पे बैठ गए. हम भी बैठ गए. अब मेरे साथ एक लड़की बैठी यंग सी, कह रही सर आप पीएचडी करेंगे? मैं कहाँ करेंगे. और वो 2 घंटे का एग्जाम ऑनलाइन, 1 hour फॉर दैट जनरल एप्टीट्यूड एंड रीजनिंग क्वेश्चन एंड 1 hour फॉर योर स्पेशलाइजेशन पार्ट ए एंड पार्ट बी. हमने बड़ा कॉन्फिडेंटली कंप्यूटर पे अटेम्प्ट किया और रिजल्ट आया तो हम क्लियर हो गए. और वहाँ खत्म बात नहीं हुई. जब हमने पीएचडी की क्लासेस अटेंड करी, कोर्स वर्क 6 months कोर्स वर्क, तो एवरी सैटरडे क्लासेस एट एमईटी यूनिवर्सिटी, वहाँ बायोमेट्रिक अटेंडेंस. और जब हम क्लास में अटेंड करने गए तो वहाँ पे जो टीचर थे, व्हेन आई वेंट टू द क्लास दे वर माय स्टूडेंट्स. तो एक टीचर बोली सर अगर आप क्लास में बैठेंगे हम तो पढ़ा ही नहीं पाएंगे. मैं कहा नो, आई एम द स्टूडेंट नाउ. अब हम पीएचडी कर रहे हैं ना, तो आप हमको पढ़ाइए. बोले नहीं सर, हमें एक ही रिक्वेस्ट करेंगे, आप आइए जरूर बट गो एंड सिट इन दी लाइब्रेरी, आप क्लास में मत बैठिए. बड़ी अजीब सिचुएशन. तो आई यूज टू कैरी माई ड्रग कंट्रोल डिपार्टमेंट फाइल्स ऑन एवरी सैटरडे टू दी एमईटी यूनिवर्सिटी. जब भी टाइम मिलता था आई यूज टू सिट इन दी लाइब्रेरी एंड वहाँ पे फाइल भी करता था और पीएचडी भी चल रही थी. एग्जाम अनाउंस कर दिए, पीएचडी के एग्जाम है जी, अब 4 पेपर, रिसर्च मेथोडोलॉजी एंड सो ऑन. और नो हॉलीडे, कोई ब्रेक ही नहीं, 4 दिन कंटिन्यूटी में एग्जाम. तो मैंने उनके एग्जामिनर इंचार्ज को बोला यार 4 एग्जाम कंटिन्यूटी में कैसे हो सकते हैं, हमने तो बीफार्म में भी एग्जाम दिया, एक दिन का तो छुट्टी होना चाहिए. बोले सर पीएचडी में तो ऐसे ही होता है. अब हमें डर लगा कहीं फेल ना हो जाए, क्योंकि क्लास तो अटेंड करी नहीं. और जो कड़ी 83 में बीफार्म किया, 85 में एमफार्म किया. बट आई टुक 15 days अर्नली फ्रॉम माई डिपार्टमेंट. और मैंने वैसे ही स्टडी करी जैसे मैंने बैचलर या मास्टर ऑफ फार्मेसी में करते हैं. 4 एग्जाम में मैं अपियर हुआ और सब में A+, A ग्रेड आया और कोर्स कंप्लीट हो गया. और चैलेंज कब आया? 2021 आई हैव टू सबमिट माई थीसिस और कोविड एंड पेंडेमिक टाइम आया. दैट वॉज द टफेस्ट टाइम ऑफ माई लाइफ. Such a challenge. I used to get 2000 calls per day on my mobile, 2000 calls per day. You name any VIP, any judiciary, any member of parliament, sir medicine चाहिए, injection चाहिए, क्योंकि डिमांड बहुत थी. There was a huge gap between demand and supply. अगर 10 इंजेक्शन आ रहे थे 100 की डिमांड थी. Such a pressure. और उधर से पीएचडी भी चल रही थी और थीसिस भी सबमिट करना था. But by the grace of God, दोनों चीज़ें निकल गई। एंड आई वॉज अवार्डेड माई PhD जस्ट 8 months बिफोर माई रिटायरमेंट इन 2021। और उसका बेनिफिट क्या हुआ? उसका बेनिफिट ये हुआ कि जैसे मैंने जॉब से मैं रिटायर हुआ गवर्नमेंट से, आई गॉट सो मैनी ऑफर्स फ्रॉम यूनिवर्सिटी, सर आप हमारे यहाँ ज्वाइन कर लीजिए। देन आई प्रेफर टू ज्वाइन SGT सिंस इट इज़ नियर टू माई रेजिडेंस एज ए प्रो वाइस चांसलर, एंड टुडे आई हैव completed more than 3 years as a PVC looking after different specialization. Thanks to Government of India, now I am the member of Drug Technical Advisory Board. जिस UPSC को मैंने फेस किया एज ए कैंडिडेट फॉर माई सिलेक्शन, proudly I am telling you now I am the UPSC advisor and UPSC expert also. And with the support of all the pharmacists, क्योंकि मेरे खून में फार्मेसी प्रोफेशन है, मैं तो यही कहूँगा आपका फ्यूचर भी ब्राइट हो। और डॉक्टर मोनिका मेरे से नाराज़ रहती है कि सर आप आते नहीं हैं हमारी यूनिवर्सिटी में, और कल भी मैं नहीं आ रहा था क्योंकि मेरी बड़ी बिटिया का कल बर्थडे था, और मुझसे नाराज़, वो भी फार्मासिस्ट है, वो भी फार्मेसिस्ट से डिस्चार्ज उसने किया, पर मैं डॉक्टर विमलेश को, डॉक्टर मोनिका को मना नहीं कर पाया क्योंकि पीछे पास में कई बार बुला चुके, मैं नहीं आ पाया। पर मैं कहा इस बार जो मर्जी हो जाए, डेफिनेटली आई विल गो टू LPU, मैडम की शिकायत भी दूर करेंगे, डॉक्टर विमलेश को भी सेटिस्फाई करेंगे, और आप सब लोगों से भी मिलेंगे। दैट्स व्हाई आई एम हियर। सो विद दीज रिमार्क्स आई विल नॉट टेक मच टाइम। और जब भी मैं किसी भी कॉन्फ्रेंस या उस पर जाता हूँ, मैं सभी बच्चों से एक चीज़ बड़ी शेयर करता हूँ, बहुत अच्छा लगता है मेरे को, कि जो हमारे अब्दुल कलाम जी थे, जो हमारे प्रेसिडेंट ऑफ इंडिया थे, उन्होंने एक कोट बोला था बहुत बढ़िया। कहना कि जब भी आप सुबह उठते हैं मॉर्निंग में, अर्ली मॉर्निंग, मोबाइल भूल जाइए, मोबाइल को मत देखिए। उन्होंने एक चीज़ क्या बोली, 5 चीज़ें याद रखेंगे, 5 सेंटेंस। सबसे पहले आप बोलेंगे आई कैन डू इट। क्या बोलेंगे? जोर से बोलिए। आप सब कुछ कर सकते हैं, यू कैन डू इट। सेकेंडली ही टोल्ड यू विल स्पीक दैट नथिंग इज इम्पॉसिबल। कोई ऐसी चीज है जो पॉसिबल है? नहीं है। एवरीथिंग इज पॉसिबल, नथिंग इज इम्पॉसिबल। थर्डली यू विल स्पीक गॉड इज विद मी। भगवान को मानते हैं? मानते हैं? मुझे लगता नहीं ये यंग जनरेशन मानती है, क्योंकि मैं भी जब पूजा करता हूँ, मेरे बच्चे कहते हैं डेडी की पूजा क्यों करते हो आप? भगवान को मानना चाहिए, बिकॉज गॉड हेल्प्स दोज हु हेल्प देमसेल्व्स। ओके, एंड देन ही टोल्ड, आप बोलेंगे टुडे इज माई डे। आज आपका दिन है कि नहीं है? क्योंकि आज आपका दिन इसलिए है, यू आर अटेंडिंग दिस इंटरनेशनल कॉन्फ्रेंस with the support and guidance of Dr. Monica, Dr. Bimlesh and entire faculty, and interacting with such a renowned galaxy of people who will be sharing your thoughts and their thoughts, for research and development, particularly animal health. क्योंकि एनिमल हेल्थ के ऊपर मैं एक एग्जांपल और शेयर करता हूँ, मैं सर से भी शेयर कर रहा था। मेनका गांधी वाज टोटली अगेंस्ट एनिमल के यूज के लिए, नहीं ये नहीं होना चाहिए, वो नहीं होना चाहिए। एंड व्हेन वी वर इन ड्रग कंट्रोल डिपार्टमेंट, लॉट ऑफ मीटिंग्स, नासा जी ऑक्सीटोसिन का मिसयूज होता है, ये इंजेक्शन लगता है भैंसेस को दूध के लिए, इसको कंट्रोल करिए। चैलेंज था, ये एग्री। सर इज द मेंबर सेक्रेटरी, वेटरनरी की सर बात कर रहे थे, बहुत बड़ा चैलेंज है। सर बात कर रहे थे CROs की, बहुत बड़ा चैलेंज है। बट एथिकली वी हैव टू सी, regulations are there, everything is there. Ethically we have to regulate each and everything, definitely success will be there. So with these remarks, my best wishes to all the young pharmacists, and I wish you a very bright future ahead. And also I appeal to all of you, at least please become the member of Indian Pharmacy Graduate Association. और एक चीज़ मैं और आपको शेयर कर दूँ, दो इम्पोर्टेंट चीज़ हैं। मैं बात कर रहा था इंडियन फार्मास्यूटिकल कांग्रेस की। 74th Indian Pharmaceutical Congress this year is going to be held at Bangalore on 19, 20 and 21st December, which is hosted by Association of Pharmaceutical Teachers of India, APTI, And next year, 75th Platinum Jubilee will be hosted by Indian Pharmacy Graduate Association. So, my invitation to all of you for 74th IPC and definitely for next IPC also well in advance. And also we are holding 37th annual conference of IPGA coming Saturday on 8th of November at Lucknow. My invitation to all of you, but that is only possible once you become the life member of IPGA. Uska ek reason hai, as on date, life membership fee is only 3,100 rupees. But from January onwards, we are going to amend this fees. It will be 5,000 rupees. So if you want to become a member and if you want to make IPGA Fagwada chapter, जिसके मैडम एंड डॉक्टर बिमलेश ऑफिशिएटिंग मेंबर्स हैं, उनको स्ट्रॉन्ग करना है, तो प्लीज बिकम द मेंबर ऑफ आईपीजीए. थैंक यू वेरी मच.
Thank you, sir, for those valuable insights and for beautifully setting the tone for the sessions that lie ahead. Now I request all our esteemed guests and dignitaries to kindly move to the front row down the stage. Dear audience, let's give a round of applause as a token of gratitude and respect for our guests as they make their way to their seats.
You know, Chesta, every year over 115 million animals worldwide are used in scientific research, a number that reminds us how vital yet how sensitive this field truly is. That's why today's session holds such great significance. It's not just about scientific advancement, but about finding that balance between discovery and compassion, where innovation walks hand in hand with ethics and sustainability. For every student of School of Pharmaceutical Sciences, this session is an opportunity to understand how responsible research can shape a more humane and ethical future in science.
Now we begin with the most awaited segment of the day, the keynote lecture session. We have with us eminent speakers who will share their thoughts and experiences, helping us gain new perspectives. The first keynote lecture will be delivered by Dr. Neeraj Khatri sir, and the session will be chaired by Dr. Amritpal Singh sir. I would now like to request Dr. Amritpal Singh sir from Guru Nanak Dev University, Amritsar, to kindly take over and introduce our distinguished speaker. Ladies and gentlemen, please welcome sir with a huge round of applause.
Very good morning to all the dignitaries and the delegates that are attending this meeting. For me, I suppose rather than this conference, we are here to discuss our vision on enhancing ethical and sustainable animal research. I think it's a more than conference, it's a sacred function for all of us. Generally, whenever we are sitting together, we normally talk about science. And in science, we only talk about passion for science. But I think here we are together to discuss passion with compassion, because that is very important, that we are generally looking at our growth, we are talking about our publications, but we leave behind, we don't discuss the miseries that experimental animals they suffer if they are not taken care of very correctly. So I think it's a right platform to discuss all these issues, and it's a privilege for me to introduce the first speaker, Dr. Neeraj Khatri. He is the Chief Scientist, CSIR, Institute of Microbial Technology, Ministry of Science and Technology, Government of India, Chandigarh. And what I know, he is one of the very pioneer members, those who are associated with animal welfare, and he's a trained veterinarian working for decades for animal welfare and animal experimentation. So I would like to invite Dr. Khatri to come on stage and share his words of wisdom with us.
Let's welcome Dr. Neeraj Khatri, sir, with a huge round of applause.
So यही पीपीटी है ना, ये सही से मूव हो रहा है मूव. It's not moving. So can you change from here? It would be nice अगर ये काम कर रहा था. So because I would like to move on. So you can start time change. नहीं कोई बात नहीं, मैं अच्छा ये तो काम ही नहीं कर रहा है, राइट? जस्ट डन. सो दिस वे मूव फॉरवर्ड. ठीक. हाँ. Good morning, everybody. First of all, I would like to thank the organizers, Professor Monica Gulati, Professor Bimlesh, and all the members of organizing committee for inviting us to interact with such a huge gathering here. So I'm very happy to be part of this event. I was very impressed to hear from Dr. Datta, Professor Atul Nasa, and with me, all of you must be very impressed listening to them. So actually Bimlesh requested me that I have to finish this talk by 12:10. So without taking much time, I would be coming directly to my talk, because this session on this SR seminar is very important, not only from pharmacological aspect, but animal welfare, and connecting them, this seminar, with the sustainability goals of United Nations. It is very important because, as I'm trying to connect this topic, sustainable science through GLP, enhancing animal welfare and infrastructure. So we have listened to Professor Datta that if good science can be done only by using good animals. If your animals are not of good quality, what results you will be getting? So little bit, I will be spending few seconds on CCSCA. Actually, this CCSCA was first formed in 1960s with Professor Nain Bajaj, who was the first chairperson of this committee. However, this committee was more or less defunct till 1999. In 1998, these rules were again framed, and it is not like that, okay, Menka Gandhi was stopping the animal experimentations, but in 1960, in that CPCSA committee also, mainly, heads of all the research institutes, they were part of that committee. But there were so many complaints that, animal cages were used to be lying in the corridors of AIMS and other many institutes without food, without water. They were, extreme weather conditions they were facing, and those animals were being used for research. So I'm thankful to the CCSCA for framing all these guidelines for the benefit of the animals, and by using animals, ultimately, we are getting the benefit as human beings. So GLP is a qualitative system, right? So this is MTech. I'm skipping because of time. So, we all should be thankful to the animals because they have been sacrificing their lives for us. I would like to share one interesting data with you that, we are sacrificing 50 to 100 millions of animals throughout the world annually, right? And we have been listening about Nobel Prizes because, every day you are listening that President Trump is asking, okay, give me Nobel Prize, right? But in terms of animal welfare, out of 235 Nobel Prizes which have been given to scientists who are using animals in physiology and medicine, you will be happy to know that out of those, 195 have been given to those scientists who are using animals in their research. So I think we should thank our animals, right? And when we are using animals, it is the responsibility of the project investigator or students who are working on the animals. So PI has to ensure that, whosoever is using the animals, they are properly trained. They should be efficient in doing animal experimentations. Like if I am not able to give injections to the animal, I am not trained in that, so your results will not be good. So we need happy animals, healthy animals for our good results. And for that, we should be aware of all the species-specific information, their anatomy, physiology, nutrition. We have to, house these animals separately because every animal, they have their species-specific behavior. Like if we are housing rats and mouse in similar room without any separation, so mouse will be under, stress because in natural condition, rats, they are predators of mouse. So like we are sitting in this auditorium and somebody is like standing in front of you with a AK-47 rifle. So we all will be under stress. So that is why we have to be very cautious when we are using animals. So like when we are discussing about the drug development stages, so it means normally we take around 10 to 12 years for a drug discovery, right? Starting from discovery, then non-clinical studies, then means non-clinical when we are using animals proof and concept, then we have to go for clinical trials, drug approval, Dr. NASA is there, manufacturing. So total, more than 12 years we have to spend on that. So like recently, we have to like FDA has stopped some of the animal experimentation. So it's an opportunity for South Asia, like all the animal studies. Now we have the opportunity to conduct in our country also. So, but when we are doing that thing, so we need to have, facilities and infrastructure, like we are discussing about sustainability goals. So one of the sustainability goal number 12 is that we have to have sufficient scientific infrastructure to support our scientific excellence. So GLP is part of the quality control system. So what is GLP? In fact, when I came last time here, so Dr. Monica and Bimlesh, they were very interested to have GLP facility in LPU because there are very few institutes in India who are having GLP, right? So that is why I thought that this time, we have Dr. Tyagi will be discussing about animal welfare and guidelines, and Puneet Bansalji will be discussing about 3 Rs, right? So this is a GLP is nothing but is the managerial concept of quality system. It is concerned with the organization process and the conditions where the studies will be planned, how they will be performed, monitored, recorded, archived and reported. Means everything, whatever you are planning to do, it should be a written protocol which has been approved by the organization. Who will be doing, whether they are properly trained or not. So everything has to be documented. So as I said that it is a quality system concerned with organization processes and conditions like non-clinical health and environment safety studies, how they will be planned, performed, monitored, recorded, and as per guidelines we have to archive all those results and then we have to report. It assures the quality and integrity of studies that are intended to support research on drug development. And it is minded, it is only a quality system. We are not worried about the scientific value of that because everything has been approved by the management. So when we are being provided with that document, we have to see whether it is being properly followed or not. So as I said, that scientific endeavor will always have its fair share of uncertainties, errors and disputes, but it is the misconduct that is impacting the results. So many times we will be reading the news that so many papers they have been retracted, right? Or some audit has failed, right? Like in Panacea Biotech, they were doing very well, but because of few WHO audits, they failed and then it came down. So it is a transparency of these regulatory processes and mechanisms for investigation allegations of misconduct that will improve the trustworthiness of science in long run. So we have to maintain the quality. So actually now question comes, why we need these GLP regulations? Because we, historically there were so many issues, like I'm just giving you brief history also. In 1937 we have this sulfanilamide tragedy. So there were so many cases of misconduct, like thalidomide tragedy, silent spring, non-clinical studies. Because when FDA, actually they investigated all those complaints, so they find that nobody was following the procedure. Drugs were being tested on humans without any clinical trial or without any approvals. So after going through all those results, then it has to be, GLP was formed. So I was discussing about this elixir tragedy also, where, mass poisoning of 105 patients was there because they were treated with an untested medication. So medication was not approved and that medicine was being tested on the patient and 105 patients were died. Similarly, this thalidomide disaster was there. So one of the scientists who was reviewing this FDA thing, she refused to authorize the use of this compound because she had concern about the safety of the drugs. So throughout the 60s and 70s, a climate of environmentalism and fear of the negative health of effects on the animal. So that was widespread in USA, and all these conditions led to the forming of GLP. So mind it, India is not a still signatory of OECD guidelines, but it is a very key, India is a key member of that OECD, but not a signatory. So these are the significant events which led to development of GLP guidelines. This is a handbook of GLP. So as I said, the purpose of GLP is to promote for generation of quality data and to provide managerial tool. The objectives are to empower disease endemic countries to perform research activities which are acceptable throughout the world. Like if we are, I have a certified GLP lab in India. So whatever data I am generating, this data is acceptable throughout the world. So that way, we need not to repeat those experiments when we are not repeating, not reproducing. So that way, we are saving lots of animals. Not only animals, we are saving lots of cost also, right? So this is called MAD, mutually agreement data. So GLP studies include non-clinical lab studies, in vitro or in vivo test. Then we are using different test articles. We are using different test system like animals which we are testing. But this doesn't include human subjects or clinical studies. This term also does not include basic exploratory studies. We can always say that, okay, we are doing any basic study as per GLP compliant. But getting the GLP certificate, we have to apply to DST, our Department of Science and Technology. There they will approve the all toxicological studies, and so these are the examples of animal studies which are done under GLP, like ADME, all toxicity, carcinogenicity, reproductive toxicity, mutagenicity, eye irritation, dermal irritation. So as I have already discussed this. So what are the fundamentals of GLP? So important thing is that what are the different parameters we have to see, what are the resources we are having, like organization, test facilities, personnel and equipments, whether we have those equipments which are required for doing those particular studies. Quality, we need to have one QAU, quality assurance unit, because they will be independently checking whether you are following proper guidelines or not, whether you are following all the rules and guidelines, because we have to have standard operating procedures for all procedures. Then whatever test items we are getting for toxicity or any other test, we need to have proper characterization of test item, test systems, reagent and materials. Then ultimately, whatever we are doing, whatever we are collecting raw data, final report, we have to archive those things. So this I have already discussed. Then like as I told you that if test data is from any lab or any country, it should be acceptable throughout the world. So it avoid duplicative testing, it break the technical barriers and cost containment also. So we have to weigh whether we require cost of quality and no cost or lack of quality. So obviously we will go for quality only. So these are the different elements of GLP. We need to have personnel, management and test facility manager. So management is very important when we are applying GLP principle in any of the institute, because management is the one who will be overseeing the requirement of manpower, how many people they are trained in desired experimentations. Then we need to have standard operating procedures. We need to have facilities. Like if I'm planning any experiment on transgenic animals and I'm not having that facility, so that way, you know, you are putting those transgenic animals under, because they are very, their immune system is very deficient, so they will be, they will not survive that environment if you don't have the facility. Then we need to have test and control article, characterization, formulation, proper SOP for storage. Then key activities to implement the GLP is, first of all, we need to have standard operating procedures. We need to develop standard test procedures, specifications of test and materials, and control of documents. So we have to, whatever SOPs we are making, so those SOPs have to be approved by the management, and these SOPs then will be given to the manpower who will be doing those experiments. Training of manpower is very important. Then post-training evaluation and quality assurance unit, they have to examine whether those approved manpower, they are doing those activities properly as per SOP or not. Upkeeping of lab with required facilities again very important, like chemical and instrument lab, microbiology, pharmacology, animal houses, because test item, when we are testing the any new chemical entity or vaccine, like everything has to first evaluated on the animals only. Then whatever results we are observing, we have to report honestly without any scientific bias. Presentation of the results as per the predetermined or pre-approved format. Rechecking of data by second person who is directly not involved in that particular study. And when we are taking readings on the equipments, we need to have calibration of those equipments regularly. So what are the test facility management's responsibilities? So we have applied for test JLP certificate and we have got the certificate. Then, you know, management has to ensure that we have sufficient number of qualified persons to do those studies, appropriate facilities, equipment and materials. We need to perform or complete the objectives of the project within the time frame, and have maintenance of personal record of qualification and their training also. So one of the important pillar of any GLP program is quality assurance unit, because they serve as our internal control. So they, as I said, that they will be independent of the study group or study personnel who are doing those studies, but they have the standard operating procedure or project proposal of that study. So they can come at any time without any prior information also, that how you are performing your experiment, whether you are following SOPs or not. So they assure management that facilities, equipment, personnel, methods, everything is as per the GLP. Planning of studies, resource allocation, they will evaluate the whether we have qualified staff or not, and organize record keeping. Then another point of conduct person is study director, who will be the responsible of that particular study. So he has the overall responsibility of technical conduct of that data, interpretation, analysis, documentation, and finally reporting of the results. Then after study director we have PI, principal investigator. PI ensures the study is conducted in accordance with the approved GLP, and study personnel responsibility are recording of all raw data as per compliance with the principle of GLP. If we find that there is any deviation from the instructions to be reported, the PI has to report it to the study director, and study director will report to the management and once management approves that deviation, then only it will be part of approved project. And take health precautions and personal safety. So it is also the responsibility of PI that he and his or her staff members who are doing experiments, they follow all the precautions. So we have discussed. Then finally, validation of analyst. So like sometimes, it is not always true that we will get whatever we have proposed. So there might be some variation also. So we have to take into account all those variations, and we need to have it is necessary to validate the analysis in terms of precision and accuracy. So it should not be variation, means we should not have SD level should not be very high, otherwise it will impact the study. So variation should not be more than plus minus 1% by different analysts. Then whenever we are doing any study, we have to follow personal safety also. Like when we were discussing about COVID, so in our institute also because we had BSL-3 lab, so we were involved in all the COVID testing samples also. But when you are handling any biological or infectious agent, we need to have proper biosafety program. So we have to wear protective gloves, all the PPEs we should call them. Glassware should not be used for whatever lab glasswares are there, we should not use for our eatables or drinking. Chemicals should not be tasted, and whenever we are pipetting, it should not be be done using mouth. And then these are the documentation requirement which every management should have. All the written documentation about organization chart, job description, CVs, training records. Why it is important? Because whenever you are having GLP certification, so management will be getting so many studies from the sponsored studies also. So unless management is knowing that how many trained manpower we are having, so we cannot have n number of studies. So that way it is very important. So up-to-date training records should be done. So whenever all the facilities I said, then most important thing is that when we are using test item like test facility, as per our studies protocol, so animal house is very important. So we need to have all these facilities for separation of facilities, isolation of individual projects. We need to have quarantine facilities, and most importantly, whatever projects we are getting, we need to have institutional animal ethics committee for approval. And so many things I will be skipping, because all those things will be discussed by Dr. Vivek also. So main thing is that we have to follow all the CCSA guidelines whenever we are managing our animal facility. So quality of animal is very important. Like in India we have accreditation of CCSA, but worldwide we have AAA LAC. These are all accreditation which means these associations, they are evaluating how you are maintaining your animal facility in terms of quality. So then finally, most important thing is that whatever results, like if you are doing any histopathological examinations, you have to archive all the data, including slides, minimum for 5 years. And when you are submitting your study report, so significance is like when you are signing the report that we have successfully completed the project, then it is your signature. So it is very important. So GLP is nothing. It is a quality assurance system that whatever we are doing studies, they are as per the approved protocols, written documents, standard operating procedures. So with this, this is all about I would like to discuss.
Thank you very much, Dr. Khatri, for this elaborative lecture. The session is open for discussion, please. I would like to have some questions from the young minds that we have in this hall.
Anything related to GLP or CCSA or animal welfare?
Thank you, Dr. Neeraj, for this wonderful talk. I think your talk is every time it adds information to our knowledge. So as you talked about GLP, there are a lot of, it is a complex process because we tried a lot to get a GLP, and if overall we see the process is too long, costly, demand a lot of manpower. So at this moment, if we can have more information about the MAT that you have talked about, that is mutual agreement of data, I think that is a possible way for the academic institutes, and I think most of the research institute also, because at CSR we have at CDRI and we have at IITR, and then the data that we need, we can get it from that place. But at our institute, if we want to have such thing, I think MAD is a way that we can follow, and if you can put more light on that, so that will add more to our knowledge also.
Yeah, you are right that, as I said, that when we are signatory of OECD guidelines, then whatever data we are generating in different labs in India, it is acceptable throughout world. So, but having said that, as you are saying that we can't take data from CDRI or something like, but nowadays it is a very simplest process. So you can, because let me tell audience also, this getting a GLP certificate is not a mandatory thing, but it is a voluntary, because it is a qualitative system only. So whatever you are doing in your lab, it is acceptable to outside people also. So once you get the certificate, so annually GLP inspectors will be coming to your facility and they will be evaluating as per the documentation whatever you have submitted. So nothing to worry about all those inspections if you are following all the guidelines. But India is still not a signatory to OECD guidelines, but it's a key member. It is official statement in OECD book also. But India is currently working on that aspect also, and I hope that very soon we will be part of that thing also.
Thank you so much, Dr. Neeraj Khatri sir, and a heartfelt thanks to Dr. Amrit Pal Singh sir for chairing this session so wonderfully.
Thank you very much.
Let's hear it for both of them with a huge round of applause. is not just an institution, it's a world in itself, a place where every dream finds direction and every student finds a purpose. LPU believes in the holistic growth of its student, not just academics, but overall personality development.
बिल्कुल सही कहा चेष्टा। लवली प्रोफेशनल यूनिवर्सिटी एक ऐसी जगह है जहां हर स्टूडेंट को मिलता है एक नया मौका अपने आप को पहचानने का, अपने टैलेंट को दिखाने का और अपने सपनों को पूरा करने का। यहाँ सिर्फ पढ़ाई नहीं होती, यहाँ लाइफ लेसन्स मिलते हैं, बीट एकेडमिक्स, कल्चरल इवेंट्स और लीडरशिप एक्टिविटीज़। लवली प्रोफेशनल यूनिवर्सिटी हर फील्ड में एक्सीलेंस को प्रमोट करता है।
Exactly, and if we talk about non-academic growth, I think all of us can agree that there's always something going on in the campus. From the vibrant One India, which celebrates unity and diversity, to Magnitude, where the campus transforms into creativity and a festival of energy. And how can we forget the freshman talent research when new students like us get to show who we really are?
और यही इवेंट्स तो एलपीयू को अलग बनाते हैं। हम जैसे नए स्टूडेंट्स के लिए ये सब एक्सपीरियंसेस बहुत स्पेशल होते हैं। और मुझे लगता है कि आप सब भी एग्री करेंगे कि जब पूरा कैंपस एक रंग में रंग जाता है, एक सेलिब्रेशन बन जाता है, तो एलपीयू लगता है होम अवे फ्रॉम होम।
And not just events, even our opportunities are endless. You know, we both are freshers, and yet within just 2 months of joining this university, we are standing here anchoring this grand conference. And that's only because of encouragement we received from our mentors and teachers who pushed us to go beyond our comfort zone.
बिल्कुल चेष्टा, क्योंकि यहाँ के मेंटर सिर्फ पढ़ाते नहीं हैं, वो इंस्पायर करते हैं और वही इंस्पिरेशन हम सबको मिलती है हर इवेंट के ज़रिए।
Exactly. LPU believes in creating leaders, not followers. It gives us exposure, opportunities, and experiences that help us grow personally and professionally. And what's even more inspiring is that along with cultural and leadership development, LPU strongly focuses on academic excellence. And today's conference, the National Conference Enhancing Ethical and Sustainable Animal Research, is a perfect example for that.
हाँ, आज के इस कॉन्फ्रेंस में हमारे साथ मौजूद हैं कुछ बहुत ही इंस्पायरिंग पर्सनैलिटीज़ जिनसे हम डायरेक्टली सीखने वाले हैं।
Like Dr. S.K. Datta sir, Dr. Atul Nasa sir, Dr. Vijay Bhala sir, Dr. Neeraj Khatri sir, Dr. Vivek Tyagi sir, and Dr. Puneet Kumar Bansal sir. All of them are pioneers in their fields, and we are truly honored to have them with us here at Lovely Professional University.
और यही तो एलपीयू का चार्म है। यहाँ हम सिर्फ बुक से नहीं, एक्सपर्ट से सीखते हैं, जहाँ क्लासरूम और कॉन्फ्रेंस हॉल दोनों ही एक लर्निंग स्पेस है। सोचिए, एक ही कैंपस में इतने बड़े साइंटिस्ट, रिसर्चर्स और प्रोफेशनल्स हमारे सामने अपना एक्सपीरियंस शेयर करने वाले हैं। क्या ये अपने आप में एक प्राउड मोमेंट नहीं है? जोरदार तालियां होनी चाहिए।
It truly is, Mridul. Lovely Professional University gives every student, from a first-year fresher to a final-year researcher, a platform to learn, perform, and transform.
बिल्कुल चेष्टा, यहाँ हर दिन एक नई लर्निंग होती है, हर मौका एक नया एक्सपीरियंस होता है, और इसीलिए हम कह सकते हैं कि लवली प्रोफेशनल यूनिवर्सिटी सिर्फ एक यूनिवर्सिटी नहीं, एक जर्नी है ग्रोथ और ग्रेटनेस की।
Beautifully said, Mridul. A place that nurtures knowledge, celebrates talent, and inspires transformation. Let's hear it once again for Lovely Professional University. एक बार जोरदार तालियां हो जाए, ladies and gentlemen. Joined by Honorable Chancellor Sir Dr. Ashok Kumar Mittal and our worthy Pro-Chancellor Ma'am, Dr. Colonel Rashmi Mittal Ma'am, very shortly. So as soon as Sir and Ma'am arrive, I request you all to kindly give them a standing ovation and applaud for them. Rise and give a standing ovation and a huge round of applause as we welcome our visionaries. Member of Parliament Rajya Sabha and Honorable Chancellor, lovely professional University, Dr. Ashok Kumar Mittal sir, and our worthy Pro-Chancellor, lovely professional University, Dr. Colonel Rashmi Mittal ma'am. Ladies and gentlemen, please give a huge round of applause for our visionaries. Your leadership, sir and ma'am, continues to inspire every student, every faculty member, and every initiative at lovely professional university. Thank you so much, sir, and thank you so much, ma'am, for gracing this occasion with your presence. Your constant encouragement and vision truly makes every event at lovely professional university special. Now I would like to invite Honorable Chancellor Dr. Ashok Kumar Mittal, sir, and worthy Pro-Chancellor Dr. Colonel Rashmi Mittal, ma'am, to kindly join us on stage with our distinguished dignitaries, Dr. S.K. Datta, sir, Dr. Atul Nasa, sir, Professor Dr. Vijay Bhalla, sir, Dr. Neeraj Khatri, sir, Dr. Vivek Tyagi, sir, Dr. Puneet Kumar Bansal, Executive Dean and Registrar, lovely professional university, Dr. Monica Gulati, ma'am. I would also like to invite Dr. Bumlesh Kumar, sir, and Dr. Navreet Khurana from School of Pharmaceutical Sciences. Ladies and gentlemen, please give a huge round of applause as our dignitaries take their respective seats.
I would now like to request Dr. Monica Gulati ma'am to kindly present a floral welcome and gratitude to our Honorable Chancellor, Sir Dr. Ashok Kumar Mittal. Please give a thunderous round of applause that it will make this welcome more greener and warmer. Now I would like to request Dr. Monica Gulati ma'am to present a floral welcome to our worthy Pro-Chancellor Dr. Colonel Rashmi Mittal ma'am. जोरदार तालियां हो जाएं. Moving ahead, it's time for a significant academic movement of the day, the launch of the abstract book of this national conference. To grace this significant movement, may I now request Member of Parliament Rajya Sabha and Honorable Chancellor, lovely Professional University, Dr. Ashok Kumar Mittal sir, and worthy Pro-Chancellor, lovely Professional University, Dr. Colonel Rashmi Mittal ma'am, and all our esteemed dignitaries to kindly come forward and proceed for the abstract book launch. Ladies and gentlemen, I expect a thunderous round of applause for them. This abstract book launch is a symbol of collective knowledge, innovation, and research excellence that this conference represents. It is a compilation of ideas, research, and innovations contributed by scholars from across the nation. It truly stands as a testament to the pursuit of excellence and knowledge. Ladies and gentlemen, a thunderous round of applause. Thank you so much, sir. Now I request all our respected dignitaries to kindly take their respective seats.
Now it's time for one of the most special moments of today's inaugural session, the felicitation ceremony, where we take a moment to honor and express our heartfelt gratitude to our esteemed guests. May I kindly request Honorable Chancellor Dr. Ashok Kumar Mittal sir and worthy Pro-Chancellor Dr. Colonel Rashmi Mittal ma'am along with Dr. Monica Gulati ma'am to kindly come forward to felicitate our distinguished dignitaries. Ladies and gentlemen, let's fill the hall with a warm round of applause as we begin the felicitation ceremony. First, we felicitate Dr. S.K. Datta sir, who is the Joint Commissioner, Animal Welfare and Member Secretary, CCSEA, Department of Animal Husbandry and Dairying, Ministry of Fisheries, for his remarkable contribution to academics, research and institutional excellence. Ladies and gentlemen, please give a huge round of applause as we felicitate sir.
जोरदार तालियां.
Next, we felicitate Dr. Atul Nasa sir, who is the Pro-Vice Chancellor at SGT University, Gurugram, Haryana, and President, India Pharmacy Graduates Association, IPGA, for its outstanding service and leadership in the field of pharmaceutical sciences. Next, we would like to honor Dr. Vijay Bhalla sir, who is Dean ICIA, SGT University, Gurugram, Haryana, and Treasurer, Indian Pharmacy Graduates Association, IPGA, for his path-breaking research and contributions towards sustainable pharmaceutical innovation. जोरदार तालियां हो जाए हमारे डिग्निटरी के लिए. Now we would like to honor Dr. Neeraj Khatri sir, who is the Chief Scientist, CSIR, Institute of Microbial Technology, Ministry of Science and Technology, Government of India, Chandigarh.
जोरदार तालियां, ladies and gentlemen.
Next, we would like to honor Dr. Vivek Tyagi, sir, who is the Senior Consultant Committee for Control and Supervision of Experiments on Animals, CCSEA. Please welcome him with a huge round of applause. And now we felicitate Dr. Puneet Kumar Bansal sir from the Department of Pharmacology, School of Health Sciences, Central University of Punjab. Let's have a huge round of applause for Dr. Bansal. Thank you so much, sir, and thank you so much, ma'am, for felicitating our distinguished dignitaries and speakers. May I now request Honorable Chancellor, sir, worthy Pro-Chancellor, ma'am, and Monica Gulati, ma'am. Ladies and gentlemen, let's hear a huge round of applause as we capture this everlasting moment.
Ladies and gentlemen, आपकी तालियां ही इन पलों को खास बनाती हैं. जोरदार तालियां. Now, to extend our gratitude and formally conclude this inaugural ceremony, I would like to invite Organizing Secretary and Convener EESAR Dr. Bimlesh Kumar Sir from the School of Pharmaceutical Sciences to deliver the vote of thanks.
Let's hear a huge round of applause for Sir.
Good afternoon, everyone. It is indeed a great honor and privilege for me to propose the vote of thanks on behalf of Organizing Committee of EESAR 2025, the National Conference on Enhancing Ethical and Sustainable Animal Research, aligning infrastructure and welfare with SDG, Sustainable Development Goals. At the outset, we express our deep sense of gratitude to our Chief Guest, Dr. Ashok Mittal, Honorable Member of Parliament, Rajya Sabha, Chancellor, Lovely Professional University. We are equally grateful to our patron, Colonel Dr. Rashmi Mittal, Pro-Chancellor, Lovely Professional University, for her valuable guidance and motivation in promoting ethical and sustainable research culture at LPU. Our heartfelt thanks to Honorable Guest, Dr. S.K. Datta, Joint Commissioner, Animal Welfare and Member Secretary, CCACA, Department of Animal Husbandry and Dairying, Ministry of Fisheries, Animal Husbandry, Government of India. We extend our sincere gratitude to Dr. Monica Gulati, Executive Dean Registrar, Lovely Professional University, for her continuous mentorship, encouragement and dedicated contribution to the development of School of Pharmaceutical Sciences. We are deeply thankful to our guest of honor, Professor Dr. Atul Nasa, Pro Vice Chancellor, SGT University, Gurugram, and President IPGA. We are also express our appreciation to our eminent guest, Professor Dr. Vijay Bhalla, Dean ICIA, SGT University, Gurugram, and Treasurer IPGA. A special note of thanks to our keynote speaker, Dr. Neeraj Khatri, CSIR, IMTech, Chandigarh, Dr. Vivek Tyagi, Senior Consultant, CPCSEA, Dr. Puneet Kumar Bansal, Department of Pharmacology, Central University of Punjab. We are also acknowledging the presence of our distinguished guest, Dr. L.D. Singla Sir, Dr. Amritpal Singh, Dr. Dharmendra Ahuja, Professor Dr. Alka Beotra, Dr. Harish Chandar Sir, Professor Dr. Nirmal Singh, and Professor Pawan Kishan, along with that Dr. Damanpreet Singh. My heartfelt appreciation goes to Organizing Committee, Faculty Coordinator, and my dear students and technical staff. Thank you so much.
Dignitaries to kindly take their respective seats down the stage in the front row, and with deep gratitude we extend our heartfelt thanks to Member of Parliament Rajya Sabha and Honorable Chancellor Dr. Ashok Kumar Mittal Sir and worthy Pro-Chancellor Dr. Colonel Rashmi Mittal Ma'am for gracing this occasion with their presence. Your guidance and encouragement, Sir and Ma'am, will continue to inspire us all and to move ahead with renewed enthusiasm.
Zordar Taaliyan.
डियर ऑडियंस, आपकी तालियां ही हमारे डिग्नेटरीज के लिए सबसे बड़ा सम्मान है, तो एक बार जोरदार तालियां हो जाए. Thank you so much, sir, and thank you so much, ma'am, for gracing this occasion with your presence.
I would like to tell our esteemed dignitaries that Lovely Professional University has always believed that true education goes beyond classrooms. It lies in innovation, inclusivity, and impact.
Once again, everyone, let's give a standing ovation to our visionaries. It was truly an honor to have had our Honorable Chancellor and worthy Pro-Chancellor with us.
I would like that lovely profession university has always believed that true education goes beyond classrooms. It lies in innovation, inclusivity, and impact. With its world-class infrastructure, globally benchmarked academic practices, and research-driven culture, Lovely Professional University has emerged as one of India's leading institutions shaping future leaders and policymakers. The university proudly hosts students from more than 50 countries, reflecting the spirit of one world, one family. Recently, LPU has been ranked 5th in India in the Times Higher Education World University Ranking 2026, standing tall amongst all government universities, IITs, and private institutions, and placed in the 501 to 600 band globally, a true testament to excellence in education, research, and innovation.
That along with the keynote lecture sessions, we also have some very wonderful and enjoying cultural showcases planned for all of you. So kindly maintain your energy because you are actually going to enjoy the cultural events which we have planned further in this conference. Will be delivered by Dr. Vivek Tyagi and the session will be chaired by Dr. L.D. Singla sir. I now request Dr. L.D. Singla sir from Guru Angad Dev Veterinary and Animal Sciences University, Ludhiana, to kindly take the session forward and introduce our esteemed speaker.
जोरदार तालियां हो जाएं सर के लिए.
Thank you so much and good afternoon to all the distinguished guests, delegates, faculty members and participants. As I am well introduced, I am L.D. Singla from a veterinary university from Punjab, Gadwasu. I am also editor to National Academy of Veterinary Sciences, India. Before we should continue about the introduction of our today's speaker, No, I will go there, no problem. So, we have I have a proud privilege to chair this session today. I would like to begin extending heartfelt welcome to our chief guest today's morning, Dr. S.K. Datta ji. Though I have little interaction with him during some of the Joint Commissioner Animal Husbandry, Government of India, and along with Dr. Vivek Tyagi ji. He is consultant in CCSEA. As far as our today's speaker is concerned, Dr. Tyagi Saab, he is MSc Geology and PhD Entomology. He is senior consultant of the Committee for Control and Supervision of Experiments on Animals, that is CCSEA, Department of Animal Husbandry, Dairying, Ministry of Fisheries, Animal Husbandry and Dairying, Government of India, New Delhi. As far as his area of work is concerned, it relates to registration of and renewal of establishments. All the establishments, those are involved in working of laboratory animals or large animals, their registration as well as renewal. He is doing all those things. He is engaged in, those are engaged in other experimental work rate of laboratory animals, and their IEC committee formation or some renewals. Also the processing of matters related to selection of nominees as well as different trainings. Those are being done by the CCSEA members, and all other administrative matters related to CCSEA. He has delivered many of his lectures in different institutes related to animal ethics and welfare. Today, Dr. Tyagi will highlight the CCSEA guidelines and critical importance of ethical experimentation on animals. As far as his presentation is concerned, he will particularly highlight the valuable investigations, authorizations to carry out the experiments and other scientific procedures on laboratory animals following the IAEC, that is Institutional Animal Ethics Committee, as well as CCSEA rulings or approvals. Dr. Tyagi will educate on animal ethics and welfare, providing essential guidance on performing animal procedures in a manner that avoids unnecessary pain and sufferings to the animals before, during, or after experimentation that are required as far as CCSEA guidelines. So I now invite Dr. Tyagi to deliver his keynote address. Over to you, sir.
Ladies and gentlemen, please give a huge round of applause as we welcome Tyagi sir.
सभी को नमस्कार. Respected dignitaries, seniors and students, hope you have enjoyed the inaugural session of the training program with the meaningful inauguration session. First of all, I would like to express my sincere gratitude towards the organizers, Madam Monica Gulati ji and Dr. Bimlesh ji, for inviting me here to discuss the issues related to laboratory animal experiments in the country. Our respected member secretary sir, Dr. S.K. Datta sir, has already set the tone for the conference by apprising you about the importance of laboratory animals in the experiments. This lecture will be about the regulations required before, during and after conducting any experiments on animals which are required to be adhered. आप सब पेन ड्राइव लगाओ, आज का डेट की वीडियो ओपन करो, ठीक है? जब तक PPT start हो, we'll discuss some issues. Since we are aware that laboratory animals are extensively being used for the experimentation on animals in the country and throughout the world. Why we are using the animals? It is understood now. The animals are being used to find out the biological basis of disease and to find out the treatments for the human beings and as well as for the animals also. It means it is very important to use the laboratory animals for the society. But some regulations are there, some responsibilities are there to use the animals in the experimentation. For that purpose, the CCAC is there and the guidelines are framed. Just to know my audience, just wanted to know that how many of us are aware about the CCAC? Just raise the hands. कितने लोग CCAC से परिचित हैं?
इसी से आगे पीछे होगा ना?
Oh, very few are there. So, why we are using the laboratory animals and why we are talking about the welfare of laboratory animals? It's about the animals or something else. ये जो हम animal welfare की बात करते हैं, why we are talking about the laboratory animal welfare? This is the first question. Member Secretary Sir, Dr. S.K. Datta Sir has already informed that we are the selfish people. We are using laboratory animals for our own purpose. Why? Because if animal will be treated in a better way and we are treating the animals properly, that will give a good result. हमारे रिजल्ट अच्छे आए, हमारे एक्सपेरिमेंट्स अच्छे से हों, इसलिए वी आर यूजिंग द लेबोरेटरी एनिमल्स. तो यदि किसी को ऐसा लगता है कि वी आर हियर टू डिस्कस अबाउट द एनिमल्स, यस ऑफ कोर्स, बट प्राइमरली फॉर द ह्यूमन बीइंग्स. पहला विषय है कि हम अपने लिए एनिमल्स को यूज करते हैं, देन वी शुड यूज द एनिमल्स इन जुडिशियस मैनर एंड लीगली. For that purpose, one committee is constituted under the ambit of one act. It is Committee for Control and Supervision of Experiments on Animals. Oh, it's a different presentation. आपने सर का presentation open कर दिया. It's second one. This one. So CCACA, the guidelines and regulatory framework, compliance and ethical considerations before using laboratory animals in the experimentations. One committee was constituted to ensure some freedoms towards the animals. If you wanted to understand what is laboratory animal welfare and what is animal welfare, we will just find out the answers of the 5 questions. 5 freedoms. Freedom from hunger, thirst and malnutrition, freedom from discomfort, freedom from pain, injury and disease, freedom to express noble behavior, and freedom from fear and distress. If somebody is just ensuring that 5 freedoms are in place, that in a layman language we can understood, okay, this animal or it in a good state. अगर हम ये 5 freedoms को देख लें तो we can observe it that okay, it's fine. But for the scientific studies, some regulations are required, the legal framework is required. For that legal framework, one committee was established under the ambit of the Prevention of Cruelty to Animals Act 1960, the CCSEA, Committee for Control and Supervision of Experiments on Animals. It is a statutory committee. Means this committee has some legal powers under the ambit of that act to regulate the laboratory animal experimentation. It's not a matter of choice कि हमको approval लेना है तो हमारी choice है, नहीं तो it's okay. No, it's a legal requirement to take the approval to adhere the principles of laboratory animal experimentation in this country. Presently, there are 19 members in the present CCSA, wherein Joint Secretary in charge animal welfare is the chairman of CCSA, and Dr. S.K. Datta sir is the member secretary of CCSA, who is administratively responsible or administratively head of the CCSA. Apart from the administration, there are 17 expert members which are working in the CCSEA as a national committee members, nominated by Honorable Cabinet Minister. The aim of CCSE is to prevent the infliction of unnecessary pain or suffering on laboratory animals. This is the basic aim. Why this committee is constituted? To ensure that the unnecessary pain or suffering towards the laboratory animal should be avoided. This is the basic mandate to constitute this committee. The committee is constituted as per the PC Act. Some duties and powers have been allocated to this committee. Means if we have to follow something, we have to follow the legal procedure, the legal framework. This is about the lecture of today's workshop. Under these duties and powers, the CCSA formulated one rule, breeding of experimentation on animals, control and supervision rules 1998, which have been amended afterwards in 2001 and 2006 as per the then requirements. Everything what is required to adhere with for conducting experiments on animals is there in the rules. One basic line is there under the provision of above rules. Anybody, any establishment which required the biomedical research, breeding and trading of laboratory animals are required to get themselves registered with CCSE and to constitute the IEC. This is the basic idea. The quick question will come, if some individual want to carry out the experiments on animals, whether they can register with CCSE? कोई भी आप बताएं. If some individual want to register with CCSE, is it allowed? No. Organizational setup is required, means establishment is required. If somebody wants to carry out, suppose somebody means after completion of your studies or during the course of your studies you want to carry out some experiments on animals, you need to go to a institutional framework, whether you are the institution in which you are studying as of now, or you will approach somebody else to facilitate the research. Means organizational setup is required. Some functions are there, the registration of animal house facilities, establishments engaged in breeding of animals and conducting experiments on animals and renewal of registrations, constitution and reconstitution of ethical committees, approval of animal house facilities, permission for conducting experiments on larger animals, and inspection of animal house facilities where animals are kept for the experimental purpose. This is the functions. And the conducting conferences, seminar, workshop, nominee training programs, and the like for awareness of laboratory animal welfare and ethics to promote the principles of 3 Rs. Recently, one new function is added in the scope of CCSE, the grading of animal house facilities, purpose-wise, categorically, like in the medical college setup, pharmacy college setup, veterinary college, research institution, and pharmacy industry, vaccine industry, and life sciences university, etc. This is a very, very important function which is included in the ambit of this act or the guidelines. Why? Because different different institutions are having a different requirements. We cannot compare some small pharmacy college with the industry. कुछ छोटे pharmacy college को हम किसी CRO से compare नहीं कर सकते हैं. Their requirement is entirely different. Same time, we cannot compare a large farm or veterinary college with a academic small institution or a CRO. So the different institutions have different requirements to fulfill and to supervise the experiments on animals. The function is added, the grading of animal house facilities purpose-wise categorically, so that the guidelines of each facility to be adhered as per their requirement. Then the maintenance of database of available known animal methods for experiments, teaching and training purpose. This is also included in the core functions of CCSA. For this, the committees are working, subcommittees are working, and we are going to prescribe the valid alternative modules to be used to reduce the animal uses in the country. The committee is already working for this. To fulfill the functions of CCSA, some guidelines are framed, and all the material, whatever I am showing to the in this presentation is available in the web portal of CCSA, the gov.in. Very, very informative website. All the material is available in the soft copy. The guidelines for laboratory animal house facility 2015. Very, very important guideline. If you want to establish some animal house facility, what are the different parameters, the different cage sizes, requirements for particular species? Before identifying the space for construction of animal house facility, one should go to this guideline. Because if you have constructed the guidelines, excuse me students, dear friends, हाँ भाई जो बात करने वाले बच्चे हैं वो मेरी एक बात सुन लें. इट इज नॉट ए वन वे बॉलिंग. ये मैं कुछ बॉलिंग करके चला जाऊंगा और उधर से कोई रेस्पॉन्स नहीं आया तो थोड़ा अच्छा नहीं लगेगा. और हम लोग दिल्ली से अपना समय निकालकर आपसे बात करने के लिए आए हैं, तो वी शुड बी इन ए इंटरेक्टिव मोड. प्लीज डोंट टेक इट अदरवाइज. अगर हम आज एक भी विषय छोड़ देते हैं तो द पर्पस फॉर अटेंडिंग दिस कॉन्फ्रेंस विल बी डिफीटेड. हम सब अपना समय निकाल कर आए हैं. टाइम इज वेरी वेरी इंपॉर्टेंट. तो प्लीज बी विद मी, एंड इफ यू हैव एनी डाउट रिगार्डिंग दिस देन इट विल बी क्लियर्ड. So if somebody want to establish the animal house facility, they need to construct the facility as per this guideline. And if somebody is not constructing the facility as per this guideline, the registration will be refused by the committee. And that person or faculty or the facility or the company is not entitled to carry out the experiments on animals. This is the impact of the guidelines. Certain parameters are there to be followed and it is required to be followed. Then guidelines for constitution and reconstitution of animal ethics committee, how to constitute this committee, it is also there. The care and management of equines used in the products of biologicals. Equine species are being used for the production of hyperimmune plasma for that purpose, because that is separate, exactly entirely different procedure and the requirement. As of now in the country 14 institutes are there, facilities are there which are housing equines, and they are using the equines for the production of biologicals like hyperimmune plasma. For that purpose separate guidelines have been framed for the specific purpose. They are not conducting any experiments on them. They are just immunizing the equine and extracting the plasma, immunized plasma. Guidelines on the regulation of scientific experiments. It is understood that experiments are to be conducted, but how to conduct the experiments? What are the basic principles? These are mentioned in those guidelines. Then, guidelines for reuse and rehabilitation of large animals post-experimentation. It is defined in 2020. As per the extant rules, it is the responsibility of principal investigator, शोधकर्ता की जिम्मेदारी होगी to maintain the animals after experimentation, to take care of the animals. And if somebody is after completion of these studies, if you will go for the further studies, UG and PG, and after this UG you will go to the PG and PhD, and suppose you will apply for the funding in some agencies, DBT or CSIR or ICAR, and in that experimentation, if rehabilitation component is there, then financial implications will come. आपको पैसे की जरूरत पड़ेगी. You should not conduct the experiments and just leave the animals after the experimentation. The funding is required, and that time the problem will come. Who will cater the requirement of that funding? वो पैसा कौन लाएगा? So somebody who is dealing with the larger animals I'll define you afterwards. If somebody is dealing with the larger animals, they should go to that guidelines. What are the different rehabilitation methods and what exact cost is required before applying to that funding agency for your proposal. It is very, very important. Then guidelines for poultry and bird facility, guidelines for experimentation on fishes. CCSEA has introduced those last two guidelines on the request of stakeholders. हमें संस्थाओं ने, इंस्टीट्यूट्स ने अप्रोच किया that there is a requirement for framing of these guidelines, birds and fishes, then the guidelines are issued to facilitate the experiments on animals. again to facilitate. Initially the idea was not there to frame regulations for the small fishes like zebrafish, madam was just discussing with me. On the requirement of stakeholders, the institutions, the guidelines for conducting experiments on fishes have also been framed by the CCSEA. Then what are the basic principles of these rules? Very, very important points. Rule A, 3A is covered about the registration of establishment for breeding of animals or trade of animals. These are the people which are not involved in any experimentation. ये वो लोग हैं जो कोई experiment नहीं करते. क्या करते हैं? केवल breeding and trading. किसका? Laboratory animals. If somebody wants to start the business of breeding and trading of laboratory animals for the experimental purpose, ये pet shop के लिए या किसी और purpose के लिए नहीं है. It is about the laboratory animals. Then they need to get registered with CCSEA. The other one which are conducting experiments on animals covered under rule 4A. Then at the time of registration, some documents are required to be submitted. The Form A, blueprint and layout plan of the animal house facility. Why I am highlighting these documents? These are very simple documents, but having a complex meaning. The Form A is entirely different module device with different certain information which is required to be specified in the website. The blueprint and layout plan of the animal house facilities is signed by the head of the institution. Suppose head of this institution will sign any document, it means he or she is taking the responsibility. अगर किसी संस्था के head किसी document को sign करते हैं तो it means वो सारी जिम्मेदारी ले लिया उन्होंने फिर. That yes, I am aware about that how we have constructed and at the basis of the guidelines the facility is constructed. ये meaning है इस layout का. हमको लगता है कि एक पेज ही तो submit करना है, कर दो. If it is not having the specific requirements as per the regulated guidelines, that will be challenged. The application will be rejected. The minutes of the 5 internal IC members, again 5 scientists or researchers from the institutions are taking responsibility. Yes, we recommend this animal house facility for registration with CCSEA. It means they are well versed with the guidelines. If somebody is recommending something, it means they are well aware, na? Then only he or she can recommend something. Then after this approval letter from the mother organizations, to know the validity and the status of that particular institution, that registration certificate is required. Then biodata is required and the fee structure, basic fee structure is required to be submitted to CCSEA. Such important documents are required at the time of registration of animal house facility. Then suppose somebody has submitted the application for registration and the registration is done by CCSEA, then the experiments are conducted as per the distributed rules. Rule 6 says, every registered establishment shall maintain a register of particulars about the animals used from day to day for conducting experiments with the number of animals, the species, age, gender and other relevant particulars. And for that purpose, different, I will not go in detail, separate proformas are there. After completing your studies, you will get the information. If you will go for the conducting experiments on animals, then you need to maintain these records, means records are to be maintained. The point is, this is the important point. Then stocking, very, very important point. Animal house shall be located in a quiet atmosphere undisturbed by the traffic and premises kept tidy and hygienic and the animals protected from the drought and the extreme of weathers. It should be taken care while constructing the animal house facility or establishing an animal house facility. Animals cages for small animals and stables for large animals shall be such that animals can live in comfort and overcrowding is avoided. Now the question will come, अभी कोई student बोलेगा, कोई भी, बीच में से, कोई भी student, anybody, आप बोलिए, yes. How to ensure that overcrowding is avoided or not? I have already explained some guidelines are there, some basic requirements are there, measurements are there. Every species, we have prescribed the basic requirements. what is the minimum cage size or the housing requirement for a particular species. And if you are going to house a particular species like rats, then the specifications are there. By matching the specifications, we can calculate whether such species is overcrowded or not. अभी किसी के लिए तो 2 भी overcrowding हो सकता है, किसी के लिए 5 भी overcrowding नहीं है. तो कंसेंसस पे कैसे आएंगे? मैडम कह रही है कि no, we are animals are comfortable. 6 animals are housed in that particular cage and they are very much comfortable. And I am saying no, they are not comfortable, madam, it is overcrowding. तो ये निर्णय कौन करेगा? How to define this? Guidelines. Set principles are there, basic requirements are there, you calculate the requirement and define a thing. No, actually 4 animals should be housed in a particular size of cage, that is their maximum housing capacity. Then after housing of animals, the permission will come. Who will give the permission for conducting experiments on animals? The research protocols on the small animals, the definition is up to the level of legomorph, phylogenetic scale of rodents and legomorphs. The animals are categorized as small animals. And such proposals are cleared by the IAC, Institutional Animal Ethics Committee, at the level of institution. Above that level, the protocols are recommended by the IAC and further submitted to CCSA for the approval. Above that level. But CCSA has already given us some exemption for the academic studies to facilitate the academic work. Suppose somebody is dealing with the larger animals and the research protocols dealing with withdrawal of or below 0.6 ml per kg body weight, means volume is defined here, blood volume. From the larger animals, frequency is also defined here, weekly basis, for the purpose of experimentation on feeding trials, breeding experiments and livestock management studies which required only small blood collection. And such type of studies are also recommended at the level of IAC to facilitate the research work, and it is applicable only to the institutions related to ICMR, ICR, veterinary and agriculture universities and the government funded institutions handling larger animals. Then composition of IAC. What is this IAC? Again and again I am saying that IAC will approve, IAC will reject and IAC will examine. What is this? IAC is a committee approved by CCSA. And it is constituted, this committee is constituted at the establishment, at the institutional level at the time of registration of any of the animal house facility. The period of this IAC is a period of 5 years. It is valid and it is required to be coterminous with the validity of registration. The composition, there are 5 members in the IAC. One is biological scientist, 2 scientists from different biological disciplines are required, 1 veterinarian is required, 1 scientist in charge of animal house facility and 1 veterinarian are required to constitute a IAC from the institute side. Any designation can be fitted under the designations provided under the act. ये जो हमने biological scientist और ये scientist from different discipline in charge animal house लिखा है, it is required as per the rules. But any assistant professor, associate professor having the basic background of biological sciences as per the qualification can be fitted under the designations from the institution. The chairperson of the committee and the member secretary of the committee will be nominated among us the 5 IC members. But one important point is here to discuss. The chairman and member secretary should be from the institute itself. You cannot co-opt somebody. आप किसी गढ़वासु से या इमटेक से आप नहीं कह सकते हैं कि that shall you kindly become the chairman of this LPU. Why? Because these designation and people are administratively responsible for the activities of IAC. कोई भी issue होगा तो they are responsible, administratively responsible. The PI, sorry, the member secretary and chairman of the IAC should be from the institution. Some institutions are there where they want to propose their administrative head who is from non-scientific or non-biological background as the chairman of the committee, then yes, it is provision is there, then CCSA has given a provision. The 6 members can be co-opted, and that person will act as the administrative chairman of that committee. वो उस IEC में administrative chairman के लिए काम करेंगे. Having a veterinarian in the IEC is mandatory for taking care and handling of laboratory animals. This is utmost required. Veterinarian. समय की सूचना आ गई है सर. The nominees. After receiving the names from the institute side, the CCSA nominate its representative, which are the nominees of CCSA, and we are having so many nominees here in this gathering, which are working on behalf of CCSA. Dr. Neeraj Khatri sir is there, Dr. Adli Singla sir is there. They are working as a respected senior nominees of CCSA. They are responsible for the particular region and on behalf of CCSA. One main nominee, link nominee, scientist from outside, and socially aware nominees are required to be nominated. That link nominee is the substitute of main nominee. He or she can attend the IC meeting if that person is not available. In addition to the above members, one specialist may be co-opted to give the expert advice, but that will not be the part of decision-making team. The advice can be minuted in the minutes, and the decision will be made by the IEC. These are the qualification of IEC members. You can have a look on the website of CCSA. I'll skip this slide due to shortage of time. Objective, yes, very important. The first and foremost objective is the experiments to be avoided whenever it is possible to do so. And how it will be decided that whether experiment is avoided or not? Iska jawab kaun dega? If I am planning some experiments and I am saying that yes, it is very much required experimentation, but madam is saying no, it's not required. Then how to justify? By a review of literature. It means before going for the decision of any in the committee, you should review the literature regarding particular experimentation, and if it is conducted, the same experimentation repetition is not required until and unless it is required to establish some regulatory principle, or it is required regulatory experimentation. The experiments on larger animals avoided when the same results can be achieved in smaller animals. Hierarchy should be taken care of. Sir, you are going to cover 3 hours? 3 hours will be covered in your lecture? 3 hours. Okay, it's fine. Then one important point, 3 hours of animal research will be covered in the subsequent lectures. I just wanted to give you a quick information about the R. The chronology of R is very, very important. First will be the replacement, then you should see the reduction, then the refinement, and it will be covered in the subsequent lectures. Animals to be properly looked after before and after conducting experiments on them. ये animal पे experiment करने के बाद उसको ऐसे ही छोड़ देने का, it is not the responsibility. Sir, I'll just take 5 more minutes.
Because this lecture is very, very important, so we can take 5 to 10 minutes more, I think so. Rashmi ma'am, will it be okay?
Yes. Thank you so much, Chairman sir. Revision in the IC can be made in the deserving cases wherever it is required in between the validity of registration. It is also permissible. The confidentiality form, very, very important point. IPR issues are there. The confidentiality form should be signed amongst the IEC member in any format. That confidentiality should be maintained at the institution level, and it should be signed by the IEC. To formulate the SOP, this SOP formulation of SOP will resolve 99% of problems. If somebody is having some SOP for particular procedure, there is no problem, no issues will come afterwards. Just get the consensus of the members and frame the SOP. हम इस प्रक्रिया को ऐसे करेंगे. Minutize the SOP, and this will resolve all the issues. And again I am telling you, the SOP of different institutions are different. So anybody can define their own SOP as per their requirement, and that will be approved in the subsequent IC meeting, and everybody will adhere the SOP. Quorum is 6 members. The minutes are required to be submitted by the institute, and the inspection reports are required to be submitted by the nominees. The chairperson is responsible for convening of all the IC meetings, and decisions are to be communicated to the PI in writing. You should not skip some decision. And if somebody is rejecting some protocol or some project, they should justify or give the reason. Due to this reason, this protocol is being rejected. Decision will be made in the IC where quorum is complete, otherwise the decision will not be taken as, that IC meeting will not be treated as valid IC meeting. Modified proposals may be reviewed by the expeditious timeline with the identified members. If somebody is very important point, and this is the very casual and common approach. If some sir is having some small suggestion in some particular project, and I am also the member of IEC, and he is not having that much time, and every member is not having that much time. So he will, or sir will just advise to sir particular PI, you please make that correction, and Dr. Tyagi will verify this, and this protocol will be deemed to be approved. In the same IEC meeting, it will be cleared. If you are having the small suggestions, that should not be repeatedly come for the again and again for the different IEC meetings. For that purpose, IEC can define, IEC can nominate somebody to check the correction, and the correction will be approved by in the particular meeting. It is already done, the suggestions. Yes, reasons for rejection should be mentioned, and it's about the nominees and the scientists which are working in the academic institutions and the different facilities. They should know about, they should be aware about the mandate of a particular organization. Then only they will be able to justify their roles in the particular facility. And regarding academics, the major issue is there in this country as of now, whether we should use the animals in particular course or not. हमसे क्वेश्चन पूछते हैं, ये हमारे पास बी फार्मेसी के स्टूडेंट्स आए हैं, इनको एनिमल एक्सपेरिमेंटेशन करने देना चाहिए या नहीं देना चाहिए, किसको करने देना चाहिए? Crystal clear solution is there. If some experiments are defined in a particular curriculum, it is allowed, and if it is not defined in somebody's curriculum, it is not allowed. PCI has already issued the circular that in UG courses, wherever the animal experimentations are required, the information to be imparted by using computer-assisted modules, they have defined it. But in pharmacology, they are asking to conduct the experiments. If it is there, then it is allowed, and if it is not there in the curriculum, then it is not allowed. Okay, then small different procedures are there which are to be related to the establishment, which are registered with CCSE. Everything is being processed through the website. I will not take much time. And some notifications are there which are very important notifications. The number of proposals in the IAC to be decided by the chairperson and the nominee with consultation of the nominee. And the institutions, all the establishment registered with CCSE should convene minimum 2 meetings in a calendar year to check the health status of animals. The minutes are required to be submitted in this performa only. This is very important performa. By using this performa, then you will be able to know the requirement of animals. अभी अगर आपके अमेठी में the requirement of animal house facility is maximum, the maximum capacity for housing of 500 mice, and you have submitted the proposals, 6 proposals to the member secretary or to the veterinarian in a same period, that person will not be able to suffice your requirement. Then this issue will be decided then and there in that committee. You are proposing means greater number of animals, and we are not able to supply or provide that number of animals in a particular period. Either one should revise the date, or we will increase our capacity. That's why this performa is required. Very important informations are required to be captured in this performa. Yes, recently we have issued one, CCSE has issued one order regarding the IAC meetings. It has been observed that the IEC members are not going for the animal house facility and they are just conducting the IEC meeting sitting in a room, then CCSA in its 135th meeting has decided that all the IEC meetings should be conducted physically and an inspection of the animal house facility of the establishment should be conducted by the nominee of CCSA prior to every IEC meeting. It is mandatory. In case non-compliance is observed by the nominee during the inspection, no research protocol should be considered till the relevant corrections are done by the establishment. क्योंकि कुछ लोगों की शिकायतें थी कि हम लोग कहते हैं पर ये तो पूरा ही नहीं करते हैं, बोलते हैं next time we will do it, but next time when we are going that again the same answer is there, sir we will do it, you just clear the protocols. Then it is decided first to clear the observations, comply with the requirements, then to go ahead. The nominee has no restriction to inspect the animal house facility at any time apart from the IEC meetings to check the compliance. If somebody is having some compliance to be checked, then they can visit the facilities. The nominee shall submit reports after every inspection to the web portal of CCSA within 7 days of completion of inspection, because timing is very, very important. Then these are the timings. Member Secretary sir has already informed to you. Now we are working in a fast track mode. These are the timeline for processing of cases in CCSA. You can have a look of it on the web portal of CCSA. The era is completely modified. Now CCSA is behind the institutions that to expedite their work. And this is all about this presentation. The way forward as a scientist and the students carrying out research on animals, we are indebted to those animals which cooperate and make our research fruitful. Let's take care of them and handle them with respect, sensitiveness, and responsibility. One small message I also want to communicate, which is very, very important in today's era. What is this? The mantra of Rigveda. Sangachadvam samvadadvam samvo manansi janatam Deva bhagam yatha purve sanjanana upasate. Very, very meaningful shloka from Rigveda, and it is very, very useful for the scientific fraternity. What is this? May we march forward with a common goal. May we be open-minded and work together in a harmony. May we share our thoughts for integrated wisdom. May we follow the example of our ancestors who achieved the higher goals by virtue of being united. Thank you all.
Thank you so much, Dr. Tyagi, for a great message and highly informative and comprehensive presentation. And now we have some time for a few questions. Any questions? Be brief to the question and tell your name and institution for the questions. Sometimes when we conduct some of the meetings, questions arise. Now maybe even nominees of the institutions, they may also be having some questions in the mind.
Sir is having
Please, at least 2-3 questions.
Sir, को एक ये mic दे दीजिए.
My question is, how do you, it's a pretty hypothetical question, how do you think that the digital tools, AI, and other digital tools can enforce the 3 Rs? Can you put it into your policy that institutions who are having courses on the 3 Rs in their subjects, that they will be preferred to give them permission for conducting research on experiments? Otherwise, these AIs will remain AI and animals, experiments will keep going.
A very good question. The discussions are going on. 2 different mindsets are there, which are discussing this issue. The one mindset is there, which is saying that we cannot modify the complex structure with the AI or the organ on chip. We cannot modify it. The other opinion is saying, why to conduct some mere experiments on a live animal when you can achieve the small results by some indicators in the cell lines. The discussions are going on. As of now, CCSE is also framed its subcommittee which is working for the valid alternative modules for the same, like means different cell lines and the on-ground chip and different small alternatives. As of now, animals are being used and required to be used for regulatory experimentations, and we cannot completely modify the animals, but the message is by using the AI and data, we can reduce the uses of animals. We cannot replace, but we can reduce the uses of animals as of now. And it is quite possible. By using the data in different methods, we can definitely reduce the use of animals.
All right, thank you.
Thank you.
Thank you, Dr. Tyagi. It was a really informative talk that you have given. So my concern is with zebrafish. I think I wrote you in 2016.
Oh.
And you asked us to frame your own guidelines. So we framed that and we are working on that.
And we have also framed that guideline.
Yes.
On your request.
For that, I'm thankful to you and Dr. Datta that you have framed that guideline in 2021. But there is a lot of modification. What I assume that is need of lot of modification in those guidelines.
In those guidelines, which is published by the CCSE?
Yes.
Okay.
Like what you said that we have to go with Form B. Basically, Form B.
That information is not relevant to that zebrafish.
That not related to that.
Please complete your question, then I'll give you the answer.
Yes.
Yeah.
So there are several other questions because if we strictly follow those guidelines, then there may be a conflict in future because like for the treatment. So you have given the 4 ways of treatment in fish. It is IP, it is intramuscular, it is orally and using some other thing. But there is no mention about the treatment using the system water because 90% of dosing that is being done on fish, it is through dissolving the drug in the system water or in the media. So if tomorrow I mention that I have followed CPCSA guidelines, but I have used some other method that is not listed in that, it may create some problem.
Definitely
but still there are several other things because a lot of people they are buying from pet shops that are ornamental, they are not wild fish. And I don't know how CPCS is going to regulate that because definitely it's not practically possible to look each and everything, but definitely there is need of modifications of those guidelines.
Yes, so step-wise we'll just take your questions. The first question is the mandatory or requisite informations are not captured in the Form B, which is there for the rodents and other species, which is not relevant for the zebrafish. So for that purpose, we are already in the process because Form B is a part of some rules. And rules are required to be, if you want to modify the rules, then again you need to go for the amendment of rules. And for that purpose, we have already initiated the process. We are going to modify the rules. And these things will be covered in the rules the Form B is a tabular information which is required to be submitted for decision of particular project. And if some information is not relevant to a particular species, since we have decided to frame, the CCSA has decided to frame that guideline on the basis of stakeholders. I already informed to you. It was not an idea of CCSA. हम लोग शुरू में इसको regulate नहीं करना चाह रहे थे, but stakeholders has approached to CCSA because IEC was facing difficulty to approve it. IEC बोलती थी कि हम किस बेस पे अप्रूव करें. It is not covered under the scope of IEC.
Yes.
To cover under the scope of IEC, we have included the zebrafish guidelines in the scope. Now the difficulties are coming that performa which is not devised for the zebrafish is being used for submission of proposals for zebrafish. That performa can be revised as per your requirement, that is number 1. Number 2, as far as if somebody is getting, always there is scope of improvement. If we are all working in a scientific system, if you have observed that some improvements or some modifications are required to be there in the guidelines, you kindly offer the suggestions through writing a letter to sir, to member secretary sir.
Yes, definitely I will.
And if you have valid suggestions, the suggestions will be discussed in the committee and will be incorporated, certainly will be incorporated in the guidelines, and if you are having the valid points to offer. So you kindly submit your suggestions and to addressing to member secretary sir, and it will be discussed in the further meetings of CCSA.
Definitely I'll contact you.
Please sir.
Thank you.
I also have small query, although time is going. Means for teaching purpose, like CCSA is registering teaching pharmacy colleges, teaching medical institutions, veterinary institutions. I'm not talking about research purpose. For teaching also, they have certain institution they are basically into teaching mainly.
Got the point.
So in that case, is there any plan of CCSEA to offer them or to give them some alternative, because I belong to a software, pharmacology software, so I'll be talking in that sense only. So means some AI tools, some IT tools to the institution, which can help them in reducing the severity on animals. So
yes, definitely CCAC is working on this front, and we are working for this subcommittee. I have already informed to you that we have included one segment to find out the valid alternative models for some laboratory animals which can be used. But CCAC cannot endorse some software because we are regulators. Now we cannot endorse that you please use that software or this software.
Normal. Yeah.
No, I'll tell you. This is the choice of the stakeholders. We will just offer, then you should use the computer assisted tools.
Yes,
then
yes.
which tool to be used, that will be decided by the concerned stakeholders. हम वो advertisement नहीं कर सकते हैं. दूसरा क्या होगा? I'll come to your point. Yes, for the small, we have decided when this mandate is there, the grading of animal house facility. Once the facilities are graded under different capacities, like academic institutions are bifurcated, then we will be able to find out their requirement, on the basis of their requirement, the separate guidelines can be issued.
Like PCI,
we cannot endorse any
I understood, sir.
This is the compulsion.
Like PCI has told that you should use computer assisted learning.
वो हम भी कर सकते हैं.
That
yes. पर हम किस सॉफ्टवेयर को यूज करना है, ये हम ऑर्डर में नहीं लिख सकते हैं.
Understood, sir. Understood. Thank you.
Thank you. Thank you. ठीक है, डॉक्टर साहब.
Thank you so much, ladies and gentlemen. We conclude the enriching session. I would like to express my sincere gratitude to Dr. Vivek Tyagi ji for his excellent presentation. Dr. Tyagi has provided valuable insights into a range of critical topics, including the importance and framework of CCSEA rules, guidelines in governing animal research, and then about role and responsibilities of members with ethical committees, particularly IAEC, practical guidelines on layout, designs and on animal house, and documentation, registration form A, composition of IAEC and so on and so forth, and discussed about latest guidelines, something regarding fishes and others. So we are highly thankful to him for sparing his time here and discussing all these issues with us. Thank you once again, Dr. Tyagi. Grateful to you. And all the...
Thank you so much, Dr. Vivek Tyagi, sir, for such an enlightening talk, and sincere thanks to Dr. L.D. Singla, sir, for leading this session. Let's have a huge round of applause for them. And now, ladies and gentlemen, before we move forward, we will now pause for lunch. We request all our guest speakers and dignitaries to join us for the lunch arrangements made in the basement of Block 29. Let this be a time for meaningful conversations, new connections, and reflection on the inspiring sessions of the morning. Kindly return to the auditorium by 2:30 for the continuation of our post-lunch keynote sessions and panel discussion. Thank you so much and enjoy your meal. After a morning filled with knowledge and inspiration, it's time to let the melody take over, because where words and music begins. And music, as they say, is the language every heart understands. So sit back, relax, and let the beats lift your spirit. Presenting before you the music band performance by the talented students of lovely Profession University. Zordar taaliyan ho jaएं.
Check, check. Check, 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 check. Hi guys, we are Kash, and the first song is called Ishq.
Are you having a good time?
Turn on your flashlights.
मसला ऐसा है कि वो भी अपने ना हुए, दिल भी गया हाथों से, वो भी अपने ना हुए, दिल भी गया हाथों से, ऐसे आने से तो बेहतर था ना आना, हाँ दिल ना सुना ने तवज्जो से फसाना, दिल का ना सुना उसने तवज्जो से फसाना, दिल का अरे वो वक्त वो बात वो ज़माना, हाँ दिल वो भी अपने ना हुए, दिल भी गया हाथों से, वो भी अपने ना हुए, दिल भी गया So thank you everyone. This was Bandcast signing off.
What an electrifying performance. A big hand once again for our amazing band. For reminding us that music isn't just heard, it's felt. Truly, that performance has added melody to our memories today.
Do visit and explore. A perfect blend of fragrance and sweetness awaits you. So thank you so much.
For this session will be delivered by Dr. Puneet Kumar Bansal, and the session will be chaired by Dr. Dharmendra Ahuja, who is the Principal Mentor, Experimental Pharmacology, Ex-Pharma Series Software, Bureau for Health and Education, Status Upliftment, Rajasthan. I now request Dr. Dharmendra Ahuja to please take over and introduce the speaker. Ladies and gentlemen, please give it up for Dr. Ahuja. पुनीत सर का सीवी रुको.
A very good afternoon to all. After lunch, we are again here, and I would like to thank lovely professional university management, especially Dr. Monica Gulati ma'am, Monica ma'am, Bimlesh sir, and I would like to thank CCSCA for collaborating for such a noble cause for prevention of cruelty on animal. Of course, IPGA is also, IPGA dignitaries were there. So we can see, like, in addition to their normal regulatory works, they are how much concerned, they are how much concerned for the prevention of cruelty on animals. And in fact, I also belong to Bureau for Health and Education, and we also work for prevention of cruelty. And in that sense, we have designed the software experimental pharmacology series from Bureau for Health and Education. You all must all be using in your B.Pharm studies. So that's a useful software being used by almost 3,000 institutions all over India. And now coming to this session will be delivered by Puneet Kumar Bansal sir, who is professor and former head Department of Pharmacology, Central University of Punjab, Bathinda. Sir is a renowned academician. He has guided various PhD students, and he has got various international travel grants, awards, member of various renowned bodies like National Academy of Science, APTI, IPS, IAN, IPGA, IBRO. So I would like to invite Puneet Kumar Bansal sir for the keynote session. Thank you.
Ladies and gentlemen, please give a huge round of applause as we welcome sir. And I would also like to request all the students present here to maintain decorum in the auditorium. All the students, please be seated. Don't move from your seats and maintain decorum here.
Hello. Hello. So good afternoon, one and all. First of all, I thanks to the organizing team, Ma'am Monica Gulati ma'am, Dr. Bimlesh, for inviting me. And I think so, you all are fresh up now after the delicious lunch and the musical fresh up. So 2 points before starting my lecture, I want to discuss 2 points that most of my slides already discussed by previous speakers and the chief guest of today's function, S.K. Datta sir. Second thing, this is T20 match for me. Dr. Bimlesh already directed me that you have only 20 minutes to complete your talk. So I'm going to start in the fast way. So my topic is the 6 R principles. As we previous speaker discussed the 3 R's, but now I adding the 3 more R's, the 6 R principles and the ethical perspectives in the animal research. This word already discussed by Datta sir, that experimental animals, those which have sacrificed their life for the betterment of humankind. Yes, this is true. All the animals used in the research, they sacrifice their lives for the drugs that used by us, not for the animals. So it's our duty, it's our responsibility to use those animals with the ethical way, so that we minimum suffering to the animals for the humankind. So why the animals are used mostly in the preclinical studies to check the effect of drugs before their introduction to the clinical trials? I think so there is not a single drug that is not tested on the animals before going into the clinical trials. Every drug passed before going into the clinical trials from the 7 to 8 years in the preclinical research. Nowadays, the animals are also used for the medical devices testing, toxicity testing, and the safety products testing. But in the educational institute, the animals also used for the demonstration purpose. But this line was deleted by me because now the CCSEA strict guidelines that you cannot use the animals for the demonstration purpose in the educational institutes. As per PCI also, MCI also, UGC also, all given the guidelines, you cannot use the animals for the demonstration purpose, only for the research purpose. So why animals in research? Because there is anatomical, physiological, and the genetical similarity with the humans. I think so this is most of the students don't know that mouse have the 85 to 90% genome similarity with the humans. So that's why rats or the mouse are commonly used for the testing of the drugs which are used by us after completing the clinical trials. They have a shorter lifespan. If we want to test any drug from the starting of the human, birth of the human to the end, it needs 65 to 70 years. But that drug we can test in the animals within the 3 years only, because rat has the 3 years lifespan. They are easily maintained and bred in the controlled environment and the cost effective. But every coin has a 2 sides. So every point has a 2 sides. There is some people which say that animals, yes, animal used need to be used in the research, but others say no, there is no need of the animals in the research. Nowadays, AI and the other tools comes which exclude the animals from the research. So the animal testing has enabled the development of numerous life-saving treatment. That's already from the decades, every drug is testing on animals. But the opponents say that's a cruel and inhuman. There are several other ways by which we can use the test the drugs. But all this point was discussed by Dr. Neeraj Khatri, that all the discoveries, all the Nobel Prize
is based on the use of animals. Then how we can ignore the contribution of animal in the research? Yes, it's very difficult to ignore the contribution of the animals in the medical research. So if someone asks how many animals are used in the laboratory experiments around the world each year, what would be your answer? It's maybe 1 million, maybe 10 million, maybe 50 millions. No, it's 192 millions. This data is only up to 2015. So look at this data. This data is also shown by Dr. Neeraj Khatri, sir. Like this is the data from the US, China, Japan, European Union, Australia, Canada, and the United Kingdom and New Zealand. And in each in specific years, it's data up to 2020. I think so the slides is missing from the downside. This data is up to on the 2021. But no such data is available for India. In India, it's approximately 1,737 animal houses that is approved by the CCSCA, both government and the private. But if we count that each animal house use the 500 animals or the 1,000 animals in a year, it may be this count is 1.7 million in a year. So this is the question asked by the animals to us. We don't smoke, we don't drive, we don't wear makeup or perfume, we don't use paints, we don't drink alcohol, we don't drop bombs, we don't take drugs. Just because you do it, why should we suffer? This is the poster released by the CCSCA long back. So the necessity of animals in research does not mean it is ethical. So animal in research must be treated humanly with dignity, but the reality is that the level of protection afforded to research for the animals is far, far less than the afford to human participant in research. Yes, if you see, there is a clinical trial and the preclinical trials for one drug, then we see that when we see that the human participants are treated properly, but animals are not treated properly for the same trial. So it's our duty to treat the animals properly for any testing of the drugs. Sometimes animals are used unethically in research in many ways. So how we can protect or stop the unethical use of the animals? That's only possible using the law, act, and the regulation. So the first law, this is already discussed by Datta sir, the first law was passed in the British Parliament in 1822. Then the Cruelty Act was passed by the UK government, and the Animal Welfare Act by the US government. This is the law, rules, and the regulation by the Indian government. The Prevention and the Cruelty Act comes into existing in 1960s. The committee of the CCSC, the previous name of the CCSC was CIPSA. That's why here is the Committee for the Purpose of Control and the Supervision of Experiments on Animals. It is changed last year by the CCSCA. So the new guidelines already discussed by the Tyagi sir. So these are the new guidelines given by the Ministry of Health and Family Welfare. They amended the rules, acceptance of the non-animal methods, reduction of the animal testing for the new drugs. These are some international guidelines. Recently, the US FDA gives some guidelines for the decrease of the animal use in the testing of the drugs. NIH funding, they are also shifting toward the non-animal use. European Union roadmap that is discussed by Datta sir, that they are phasing out the animal testing for the chemical safety assessments. So this is how we can use the animals in the research ethically, prevent undue suffering, morally acceptable experiments, clear, rational, reasonable explanation, minimizing number of animals, skilled personnel, pre-approval of a pre-animal use, humanly treatment, minimizing pain and discomfort, anesthesia before any surgery or the post-operative care, ethical treatment of the animals. So now comes to the 6R principle. These are the 3 R's that is originally given by the Russell and the Birch, the main replacement, reduction, and the refinement. But after that, the 3 more R's was added rehabilitation, responsibility, and the reuse in that. So why do we need our concept? The concept of alternative in research, teaching, and the testing, conduct or performing more humanly animal research, play a significant role to reduce the animal suffering, enable the efficient and effective use of animal with appropriate number. So this is the historical background of the 3 R's. This R concept was given by two scientists, Russell and Bruch, in 1959. They published one book, The Principle of Humanly Experiment Techniques. In that book, they give the R concept. This is the advantage of the R concept, providing the safety, ethics, quality of research, reduction, add quality to research, reduction in the quantity, minimizing pain. So this is the first R, that is the replacement. Method that permit a given purpose to be achieved without conducting experiment or other scientific procedure on animals. A replacement means, can we replace our experiment with the other alternative method without using the animals? Yes, there are several methods of replacement of the animals in the research, like the in vitro techniques. Nowadays, the in vitro techniques are more efficient or more used by the scientists also. Before going into performing the in vivo experiment, all the drugs testing in the in vitro or the cell culture, invertebrate animals, microorganisms, using of the non-living system, chemical techniques, physical and mechanical system, use of computer stimulation. This computer stimulation means the in silico studies or the network pharmacology. So in vitro techniques, there are 3 types of techniques. One is the cell culture, cell suspensions, tissue culture and slices, and the organ culture. Recently, the IIT Madras established one lab, that is organoids labs, and they are going to start soon. They use the organoids for the testing of the drugs. So if we use the cell culture, like if any chemistry student synthesize 10 compounds or 20 compounds in their lab, in his lab or her lab, they test those compounds in the cell culture. After conducting the cell culture studies, if they find that this one compound is best, then they go for the in vivo studies. But in the previous practice, all that compounds were testing on the animals. That is the wastage of the animals and unethical practice by the PIs. Like the invertebrates, we can use the invertebrates as the alternative for the animals in our research. Microorganism, we can use the bacteria, viruses, and the fungi, protozoa for the research, like the penicillin discovery. It's not discovery, it's a serendipity. We all know that is by chance discovery. It's on the bacteria on. This is the computer-aided drug design system that is commonly used nowadays by the many scientists or the PIs, in which we can use the QSAR activities by which we can screen the number of compounds immediately. This is the flow diagram which show the discovery of from the identifying the disease, isolation of the proteins, then through the high throughput screening we can screen the compounds immediately, thousands of compounds immediately, then we go for the virtual screening and find the lead molecules. Then we synthesize those lead molecules in our lab and go for the testing into the preclinical setup. So network pharmacology also used nowadays for the other alternative for the animals, in which one drug, one target is now shifted to the multi-target, multi-pathways theory. So with this, the second R is the reduction. Method of obtaining comparable level of information using fewer animals in scientific procedure or for obtaining more information from the same number of animals. So this can be done by the animal sharing, like the control animals or the per se animals or the sham animals we can share with the other protocols, utilize animal as their own, gathering a maximum amount of information from each animal. Like if one student working on the kidney or another student working on the liver, that we can use the liver or kidney from the same animals. Consulting with the statisticians to ensure that no experiments are duplicate. This is most important because most of the students nowadays, they did not search the literature and use the same drugs for the same models again and again. So if we search the literature properly, we can avoid the duplicacy in the research. Use the appropriate species of animals so that the useful data is collected. Post-marketing surveillance. So this already shared, we can share the groups with the others. The third R is the refinement, method that minimizing pain, suffering, and the distress, and which enhance animal well-being. So refinement is very important. How we can include work for the refinement, identifying pain and the distress and the making plans for relieving it. Proper use of anesthesia, proper use of analgesic, setting the earliest possible endpoint for the experiments. So receiving adequate training prior to the performing a procedures. Yes, this is the problem with the master students sometimes. When they are not adequate trained before performing the surgery, the mortality of the animals seen, the death of the animals seen during the surgery. So before going into the surgery, you must trained under your PI or your mentor, in ensuring that doses are correct. Performing surgery and procedure aseptically to prevent the infection. The post-operative care is must for the animals. So how we can decrease the method of the refinement? Decrease invasiveness, improved instrumentations, improved control pain, improved control of techniques. So this is the best method of the example of the refinement. This is the two instruments. That is the invasive method to measure the BP in the rats, and this is the non-invasive method. I think so. My senior colleagues are here. They know that in the previous time, I think so 20 years or 30 years back, to measure the blood pressure of the animals, we need to cut the carotid artery. And we fix the cannula there, then we measure the blood pressure of the animals. And out of 10 animals, 5 died during the experimentation. But nowadays, a very simple tail and cuff method is there, in which you have to put the cuff on the tail of the animals and just you measure the blood pressure. So using the non-invasive method, we can decrease the level of pain in the animals. So microelectronics, fiber optics, laser instrumentation, the potential for refining the techniques. We can use the new techniques. Already in the market, there are several new instruments that give the less pain to the animals, reducing the level of restraint, use of the catheters, electrodes in the variety of species to allow continuous access to the various organ system. So analyzing samples, collection of the samples also improved, using the proper anesthesia and the analgesic before considering any experiments. The next R is the rehabilitation. The fourth R, now it's different from the 3R concept. The fourth R is the rehabilitation. This is also included by the CCSA. It is a process of restoring animal to a normal, healthy, and sustainable life following captivity, experimentation, abuse, neglect, and the medical intervention. So this was seen in the decade back that after, in some experiments, the animals are not sacrificed at the end of the experiments. And they kept as such in the animal house, there was no care. But after receiving the complaints, CCSA issued the guidelines that the animals which are not sacrificed at the end of the experiment need to be rehabilitated. So till the death of the animals. So it is the rehabilitation means that you have to care the animals after the completion of the experiment till the death of the animals. After care and rehabilitation of animals, post experimentation, all researchers using experimental animals have a moral responsibility to the animals after use. So this is the rehabilitation case. I think so some animal houses or the mostly animal houses have the rehabilitation cases in which the good environment is given to the animals which are not under any experimentation. Okay, the concept of fourth R, that is the rehabilitation born out of an urgent need to provide relief and succor to the aligning animals in the laboratories. Rehabilitation is the benefiting moral continuing of the third or 3R concept. During the period of rehabilitation, the animals should not be subjected or exposed to any type of activity. So this is the fifth R, that the reuse. Reuse of the laboratory animals refers to the using them again in the same or the different protocols after the completion of an experiment if an used animal equally suffices for the objective of the second or subsequent use. Yes, we can use the animals for the next experiments, but you have to take care few points. First thing, that the drugs given in the first experiments properly wash out from the animals all the organs of the animals. Second thing, the first drug did not cause any permanent damage to any organ in the animals. So after proper washing out the first drugs, all the toxins, drugs, or you can reuse those animals for the next experiments. So rehabilitated experiments, animals subjected to hepatic experiments can be reused after the completion of the hepatic clearance of the drugs. After rehabilitation for the demonstration purpose. So the sixth R is the responsibility. The monitor the animals and their wellness in the capitated condition, enhancement animal welfare, and in doing so improve the quality of science, complying with all the legal and ethical regulation and policies. Like Tyagi sir discussed that if there is no guidelines, we cannot take care of the animals. So we have to follow all the rules, regulation given by the CCSCA or the IAEC. It's our responsibility as a scientist, as a principal investigator, as a teacher, to take care of the animals which are going under any procedure in the experiments. And as a student, this is your responsibility to visit the animal house daily, twice a day, and check the animals. Recently, I am the in charge of the animal house in my university. Recently, मेरे साथ एक ये problem हुई. Problem क्या, एक complaint हुई मुझे कि कुछ students हैं, वो एक दिन first day आते हैं, छुट्टी होती है, Saturday Sunday off होती है हमारी यूनिवर्सिटी, तो वो Friday आते हैं, feed डाल के चले जाते हैं, पूरा fill कर देते हैं केज को, और 2 दिन animals को देखते नहीं हैं. तो मैंने बच्चे को, मुझे एक ही बच्चे की कंप्लेंट हुई, मैंने बच्चे को बुलाया, मैं कहा भी आपको अगर मैं 2 दिन का खाना आज ही दे दूं और फिर आप क्या करोगे. तो इसलिए कुछ स्टूडेंट्स हैं जिनको ये चीज़ें नहीं पता. इट्स ऐसा नहीं है कि वो एनिमल्स हैं, वो बोलते नहीं है तो हमें उनकी केयर नहीं करनी, तो वो भी लिविंग है, तो वी हैव टू टेक केयर एज ए लिविंग थिंग. जैसे ह्यूमंस की हम केयर करते हैं, अपने पेरेंट्स की केयर करते हैं, अपने बच्चों की केयर करते हैं. इट्स अवर रिस्पॉन्सिबिलिटी टू टेक केयर एनिमल्स विच इज विद यू फॉर योर एमफार्म थीसिस, फॉर योर डेजर्टेशन, एज योर फ्रेंड. तभी उनकी प्रॉपरली केयर हो पाएगी हमसे. So the responsible use of animal in bioscience research is essential to scientific progress, but it must always be conducted with compassion, with transparency, accountability. By following the 3 R's, adhering to legal regulation and pursuing alternative, researchers can ensure that animal use is justified, humanly, and minimized. The responsibility research is not merely about scientific outcomes. It reflects our ethical duty to respect life. So the new addition in this R is the 7th R is the relook. It's our responsibility to relook all the R's so that we can treat the animals ethically. So this is the conclusion slide of my presentation that promote the ethical research, reducing suffering, support human practice, improve accuracy, human-based methods such as organ chip provide more reliable data to human biology. So if we can alternate the animals procedure, first use the in vitro method, network pharmacology, in silico method, If you got any lead molecules or the good result, then go for the in vivo experiment. Just complete your M-Pharm, don't use the animals in your research. Sustainable and the scalable, then reduce cost and time, support regulatory compliances, future-oriented, and the encourage innovations. So with all, I end my talk with the greatest, one quote by Mahatma Gandhi ji, the greatness of the nation can be judged by the way its animals are treated. Thank you. Thank you.
Thank you, sir, for this valuable presentation. And I think this was the presentation which was most related for the sufferings of the animals, because earlier Dr. Tyagi sir had shared about regulatory guidelines. That was very important, and then other speakers are also were sharing their domain expertise. But here sir has especially emphasized, as you all are students, mostly students are here, so we should take care, we should be ethical enough, we should like Mahatma Gandhi has also told that the nation's greatness is evaluated how the animals are being treated there. So in addition to sir's note, I would also like to request you that treat animals like your child's or like your parents. Thanks.
Thank you, Dr. Puneet Kumar Bansal, for sharing such valuable insights, and gratitude to Dr. Dharmendra Ahuja, sir, for conducting this session. A warm round of applause for both of them. I would like to tell our esteemed dignitaries that lovely professional university has always believed that true education goes beyond classrooms. It lies in innovation, inclusivity, and impact. With its world-class infrastructure, globally benchmarked academic practices and research-driven culture, LPU has emerged as one of India's leading institutions, shaping future leaders and change makers. The university proudly hosts students from more than 50 countries, reflecting the spirit of one world, one family. Recently, LPU has been ranked 5th in India in the Times Higher Education World University Rankings 2026, standing tall amongst all government universities, IITs, and private institutions, and placed in the 501 to 600 band globally, a true testament to its excellence in education, research, and innovation. Ladies and gentlemen, please give it up for lovely professional university. Under the visionary leadership of Honorable Chancellor Dr. Ashok Kumar Mittal sir, LPU has become a hub of innovation and research, a place where ideas are not just imagined but realized. From groundbreaking patents to international collaborations, from youth-led startups to community impact initiatives, LPU continues to set new benchmarks in higher education. It's the dedication to progress and purpose that makes lovely professional university not just a university, but a living ecosystem of knowledge, creativity, and global transformation.
The panel discussion. This session brings together eminent experts from diverse domains who will share their insights, experiences, and perspectives on the theme of today's conference. We are truly honored to have with us our distinguished panel members. Professor Dr. Alka Biotra, Global Anti-Doping Scientist, ISO WADA Standards Leader and former Director WADA Labs, India and Qatar. Dr. Damarpreet Singh, Principal Scientist, CSIR, Institute of Himalayan Bioresource Technology, Palampur. Dr. Harish Chandra, Deputy Director, Quality Control, National Institute of Biologicals, Noida. Dr. Nirmal Singh, Professor and former Head, Department of Pharmaceutical Sciences and Drug Research, Punjabi University, Patiala. Dr. Pavan Krishan, former Professor and former Head, Department of Pharmaceutical Sciences and Drug Research. To moderate and lead this stimulating discussion, we have with us our esteemed session chair, Dr. Neeraj Khatri, Chief Scientist, CSIR, Institute of Microbial Technology, Ministry of Science and Technology, Government of India, Chandigarh. Sir, we warmly welcome you all and now hand over the session to you to take the discussion forward. जोरदार तालियां हो जाए हमारे पैनलिस्ट के लिए.
I once again welcome Professor Dr. Alka Biotra ma'am, Dr. Damarpreet Singh sir, Dr. Harish Chandra sir, Dr. Nirmal Singh sir, Dr. Pavan Krishan sir, and our session chair, Dr. Neeraj Khatri sir. Ladies and gentlemen, please give it up for our esteemed guests.
जोरदार तालियां हो जाएं गेस्ट के लिए.
I would like to call Prableen ma'am on stage to conduct the session.
A very pleasant good afternoon to everyone present over here. I am Prableen Kaur, a student of pharmacy at LPU. And it's an honor to moderate this panel discussion at the conference EESAR 2025. And I'm delighted to begin our discussion with our esteemed panel of experts. To begin with, Dr. Alka Biotra. Ma'am, I would like to ask, how does doping research in animals raise ethical and welfare concerns in pharmacological studies?
Try to let Yeah. First of all, a very good afternoon to all of you. Before commenting on anti-doping science and ethical guidelines, I would like to thank the management of LPU for organizing national seminar on such an important subject, and of course for inviting me to be part of it. Thanks to Dr. Monica and the team for this invitation. Coming again on to the ethical guidelines, I feel to really give some brief about anti-doping science. Do you all want to listen about that? It's a question from the students. Do you want to know what is anti-doping science? So that's anti-doping science is a multidisciplinary field involving scientists from chemistry, biochemistry, pharmaceutical sciences, pharmacology, like pharmaceutical sciences and life sciences. Doping is common in human athletes and animals. Human athletes, they do on their own. Animals, we all are responsible to make them dope for our own benefit. Horse is not asking that give me dope, I want to run faster. It is always the rider or the others who are insisting, who are making him take the medicine. So that is the inhuman regulatory framework for both human athletes and animals. A stringent regulatory control mechanism is there, but it is different. So I would like to cover various aspects into it, of course within a time frame, not affecting others. In human doping, doping is prevalent since ancient time, but the regulatory framework started in 1967 when the first list of prohibited drug got approved by International Olympic Commission. That time the list contained only 40 to 50 drugs, which were narcotics, stimulants like amphetamines and all. And then the testing started by IOC to test these drugs with later from 67 to now, that list has grown to 600 plus drugs and metabolites. And because the athletes and the chemists or the pharmacologists who are helping these athletes to dope, because it is both sides, more and more drugs are coming into the list and regulators are putting them into the banned list. The list has grown. Work for the laboratories have increased to develop methods, and the involvement of research to develop those stringent methods have come. Now, coming to what are the ethical, the human doping is governed now, which was earlier by International Olympic Commission, now is monitored from 2004. It's by World Anti-Doping Agency. The reason being, IOC is the one which is doing management of sports events. So they cannot be managing testing of athletes. So to give independence, to give a better regulatory framework, this was given to WADA, which is governed by both private and government bodies across the world. There are, at the moment, 29 WADA accredited labs. India has one. Asia is having 6 such labs. I had the privilege to set up the lab in India, and after retirement, I joined I was invited to Qatar Anti-Doping Lab and did 8 years over there. So the guide these WADA labs are following are as per WADA international standard for laboratories, which keeps getting updated. There is a code of ethics for doing research, for following the guidelines into research, which involves both animals and human studies.
That was very informative explanation, ma'am. Building on the same line, I would like to ask, how can research on doping agents be conducted responsibly while preventing misuse in sports or animal trials?
Now, coming to, first of all, I'd like to say, if there are 29 anti-doping labs, like there is no animal experiment is done in more than 50% of the labs. They are not doing. There are very few labs which are doing those animal studies, and the information out of that comes. In India, in Delhi lab, like from 1990 to 2016, I was there, we didn't conduct any animal studies because the animal study comes into for the methods like gene doping, for biomarker studies, for some metabolism, like later we have done in Qatar. So for some metabolites where new metabolite of a particular drug is to be identified, we can't give that drug to the human volunteers, and animal studies are to be done, which are done even like what we did was in skid mice. So humanized liver, where if a drug is given, you will get the same metabolism as in the humans. So that's what was done. And otherwise, for the methods to identify biomarkers through gene therapy in animals and by doing transcriptomics, proteomics, metabolomics, and finding the indirect markers which would help into the detection of doping agents and later used as the athlete biological passport management. So that is the thing. This is how it comes. But wherever animal studies are being done, it is in collaboration with the universities who are having proper ethical setup. They have their, because most of the labs, they are either attached to the university or to the medical college. There are few, like the Indian lab is an independent entity, Qatar lab is an independent entity, so they need to have collaboration with the institute who is having their own institutional ethics setup. So that's how we did in Qatar. And in India, the setup is mainly about human studies. And how human studies, I think, like in my so many years of experience, like for doing some metabolite testing, ethically, I shouldn't be saying at this forum, but we have taken single dose of drugs and done the metabolism of that particular drug to identify those metabolites and to keep that urine sample as a positive urine sample for reporting of any positive sample. So most of the work is, and later we took approval also for ethical to have volunteers who are taking, which is a single dose administration, which was permitted. So animal studies were not required for that purpose.
Thank you so much, Dr. Biotra ma'am, for sharing such thought. Now, moving forward to Dr. Damanpreet Singh. So my first question is, what are the most common ethical challenges observed in preclinical animal research in India?
So before I start responding to your question, I first like to thank the organizer, especially Dr. Gulati, Dr. Bimlesh and all other organizers, those who have called me here and given this an opportunity to interact with all of you, getting your views, sharing my views, and going toward your question, that is, what are the ethical challenges? I think the major challenge that we have faced earlier also, and in future that is going to be more grave, is the training of the manpower, the trained manpower. So either it's clinical, either it's preclinical, either it's in vitro, whatever it is, we need second line of researcher. They has to be trained. Like if I take example, my teachers are sitting here, Dr. Pavan and Dr. Nirmal. I think they have worked on dog. If I'm not wrong, I have just seen that equipment in my lab. So they have worked on that lab. And I cannot think that we can do work related to preclinical research using dog. But I have worked on rat. And I am lucky that I got a chance to work on rabbit also. But what about my students? I think they haven't seen rat in the lab. Those who have completed B Form, either they are doing M Form first year, they haven't seen rat. But it's true because it's even not ethical that just to train manpower and we have to sacrifice the animal, we have to chain the animal. But there is a need of certain mechanism by which we can train the next line. Because if somebody will ask me to test or to find out the jugular vein in the rat, kabhi dekha hi nahi. I haven't seen, but I can do it in rat. But my student will never see the rat. Now that is a major challenge. We need a trained manpower. And secondly, what I think that some more regulatory framework is needed to get quality animals, because in many of the cases, even Dr. Puneet was discussing that we start the experiment, 5 animals died, and from 10, 5 animals dies, and we work on only remaining 5. So this is because major case is that we won't get the quality animals. So first thing is training, second is the quality animal that has to be made available, and third thing is to have a center of excellence in each part of the country. I think even each part of the state, at least there should be at least one center. Because we say in pharmacy we get 60 students. And from those 60, I think 10 or 20 maximum, they goes to higher study and they worked on animals. So from those 20 students, those who have worked on animals, only one will proceed to further research. I think to train that one in each institute, from each state at least, we should have at least one center of excellence in every state, so that the ethical issues and all those trained manpower and what is required can be taken care of that.
So that was very informative. Lastly, I would like to ask how experiments on fish can replace animal use.
So I think I must correct your question. Fish is also an animal. We cannot say that fish as an alternative. But definitely there is again confusion. Because I'll give you my example. So in 2013 and 14, I joined as a scientist. And the major task was that our lab, we work on screening of bioactive metabolites. We take a plant, we screen it, and we get 20-50 molecules. And ethics committee will never allow to go for screening to find out one. I have to have some different mechanisms. But the problem with the natural product is that they won't enter into the blood as it is. Whatever we do in vitro, whatever we do, it won't happen until and unless we see what exactly is happening inside the body system. Because the molecule from natural origin, they have a big structure. We don't know how they are entering into the blood and in which form it is targeting to a target. For that, we need a system. And our seniors, they have asked me to establish an alternative lab. Now again, what is alternative, we don't know. So at that moment, we were having 3 alternatives. We were having zebrafish, we were having C. elegans, and third we have Drosophila. But natural product cannot be fed to Drosophila. C. elegans, but it again up to certain extent. So we choose to go with zebrafish. So being a pharmacy student, I think in India, all the courses, all the the academic degree, they won't train any person specifically to zebrafish. This training is only with, I think in fishery department or in zoology, a specific part is there, but that too not related to testing. So I was not having a test experience in that, so I took an experience from CCMB. So then we started laboratory in our institute. So the first question was, again, what is alternate? You have said that this is alternate. So kuch bhi ho sakta, butterfly can also be an alternate, but we cannot use it for drug testing. But thanks to CPCSA in 2019, they come with a they have communicated to all their establishment that these are the 5 categories of organisms that can be considered as an alternative. The first one was zebrafish. So it clears the dust that zebrafish is an alternative. So now if we see the zebrafish, again there is a need of trained manpower. बहुत ज़रूरी है. I think rat mice, though still we have seen, but zebrafish, the concerns are a lot. Because I myself have visited, I think we are in touch with all the organizations, those who are trying to start the fish facility. I think Dr. Bimlesh, he is about to start training for zebrafish at IHBT. Even to Dr. Puneet, we have provided training to their student also, to ISF Moga also. In city on 25th, we are having a same session for zebrafish. So we are trying to support a lot. I think myself, I have given 30 to 35 lectures all over the India to promote the use of zebrafish. Because whatever people are using, they are not true zebrafish. They are procuring it from, I say, Calcutta. So they are ornamental fish. So what we observe, like I am interested in studying some expression. So for that expression, I rely on databases like NCBI, ZFIN to make primers, for primer designing. But I am designing for primers for that organism which exactly not exist, because those are mutants. And I don't know how people are doing this work. There is a definitely need of certain training. I think regulators are with us. They should plan something so that at least whatever it is being used in India, it should be correct. Because even the photograph, whatever they give in their publications, that is not zebrafish, because zebrafish is a denio rareo. Whatever they are using, it is a leopard denio. It has dots. It won't have a single correct stripe. So this type of training is, I think, it's very must.
Thank you so much, sir, for your practical perspectives. Now, let's take our discussion towards another key aspect, Dr. Harish Chander. Sir, my question is, in what ways can digital tools, AI-based surveillance or data-driven approaches improve animal facility management?
Okay, thank you for your question. Thanks to Gulati Madam for invitation and her team. And thanks to Lovely Professional University. So in any animal facility management, we need to have all those animals in the animal house. So to take care of those animals, either using the digital tools or by employing the employees in the animal house is a must. We should take care of them. So right now, in most of the animal house all over the world, like, right now, the, employees go into the animal house, they take care of their bedding, they take care of their, diet, they take care of their water, or they take care of their medications if there is any needs to be given to the patients. Those are animals, right? So, and in an animal facility management, like, we need to have a certain tools for which we can, like, manage those animals. For example, like in our stables, we can have a GPS systems, GPS chips, so that we can track the movement of animals. Or if there is a time of giving a dose, medication dose to the animals, there should be an alarm, and that alarm should be on your phone, that now it's a time for giving a dose of medication to the animals. Similarly, regarding the nutrition management or the health monitoring also can be done by AI or other digital tools. And we have to have a data for that to get to that point. So like the data, we have to have a data from all over the world. Probably we have a large amount of data, and that data needs to be analyzed before we started implementing into the system. So for that purpose, we are talking of all those things now, started now, but that will take shape really into the real picture only after many years from now, because analyzing that data is not an easy job. We need to have, as Dr. Damanpreet has said, that we need to have a trained manpower. So many of the engineers or a computer scientists, they will have those degrees, BTech or MTech in data analytics, right? And they will have experience, and they will analyze that data which is collected from all over the animal houses in the world or in a particular area or about particular species. So that is how only we can take the take care of animals in the animal facility management.
Sir, that gives a strong sense like how technology is reshaping research environment. Thank you, sir. To move to Dr. Nirmal Singh for the next set of questions. Sir, my question is, what can be done to ensure that Animal Ethics Committee are empowered and impartial in their decisions?
Okay, thank you for your question. Let me first thank and congratulate Dr. Monica Gulati, Dr. Bimlesh and team for inviting me and for organizing such an important event on hot topic. And congratulations you, ma'am, and the whole team. Now coming on to your question regarding empowerment and how the ethics committee can be impartial. So there are 2-3 points which can justify this question. One is legal setup. The committees must be a part of some legislation. They should be strictly under some regulatory authority like that we have CCSEA with us, that it's a part of a law. So there should be a legal framework for the ethics committee. They should be part of this legal framework. And second, they should have clear-cut, defined goals, duties. They should be able to approve the projects. They should have power to suspend or modify. They should have the power to terminate or suspend the projects. And second point is there is a transparency in the members of the committee, and there should be a balanced composition. Members must be from multidisciplinary subjects like that we have in our IAC. There is a veterinarian, there is a person who is a social activist, various members from other biological disciplines. So in that way, there is a balanced approach, and they can justify. At second, there should be some term fixed that we have. Our Animal Ethics Committee term is 5 years. So every 5 years we renew. And our Indian system that already it is discussed that we are the best one who are following the guidelines of animal ethics. Ethical regulation in India are one of the best in the world. That is already conveyed to us. Then there is a transparency, impartialment. The members or committee members, there should be some conflict of interest. Like if a member he is presenting his own project in the committee, so he must declare conflict of interest that he will not be a party of this project. It's to be independently discussed by the other members. Then third is institutional support. Institutional must abide by the guidelines of the committees, which are properly issued, and then there should be direct directions from the head of the institutes that you are to abide by. That culture should be created. Culture should be created in such a way that we are to follow the ethical regulations, and we are to look for animal welfare, and then we are to not to compromise quality of the research. So, and one more thing, there should be some system for challenging the decisions of the committee. Means a person, there should not be any kind of internal biasness. So there should be some system for appealing to the decisions of the committee. Then there should be some external auditing as well on the decisions taken by the committee. So in that way, we can not only empower, but we can be impartial. We can be impartial as far as approval of the projects is concerned. So thank you so much.
Thank you, sir, for sharing such valuable and balanced knowledge. Now let's take the discussion towards the another key aspect, Dr. Pawan Krishan. So my question is, what are the major challenges institutions face in meeting CCSEA and IAEC Institutional Animal Ethics Committee norms?
Okay, thank you. So first of all, a very wholehearted thanks to Madam Monica Gulati ji, Dr. Bimlesh, and management of W Professional University for giving me opportunity and inviting me for on this occasion of this wonderful and very important conference or workshop, whatever we call it as, because it's a very important, which is relevant with our scientific research, basically. Because when we talk of scientific research, it has to be credible. So there is a lot of component which is required with respect to animal research. So thank you, ma'am, once again, and congratulations to all the organizers and team. So coming to your question, although it's a tough one, I think, because challenges are many, although CCSEA has laid down clear-cut directions and guidelines. So if we are following it and adhering to it, obviously we have to follow it, but some challenges when we face is like, as discussed with Dr. Nirmal also, that sometimes there can be biasness with respect to decisions, some possibilities occur, or sometimes there is a issue of conflict of interest. So some type of aspects can be there, or sometimes delay in the decisions or conduct of meetings. So some type of factors which can play a role or challenges can be faced. And one more important factor which I feel is uniformity in the standards among the animal houses. Although there are guidelines laid down by CCSEA, but I feel that there should be a uniformity, or there should be type of system to have an independent audit by CCSEA, irrespective of any academic or internal or members, like, so that as in the morning also I can remember Dr. Khatri has mentioned regarding uniformity of data generation or data acceptability. So that data, whatever we generate, if it is not, I mean to say it is not acceptable mutually across the globe, then that leads to the credibility of our research, which is not reproducible. The research should be reproducible. That is very important, because many times we see the results are varying from lab to lab. So to achieve that objective, like uniformity and independent audit, some type of steps should be taken to avoid these type of challenges in the institutions. I think that is my component.
Thank you, sir. This gives a clear view of the issues faced by the institutions. Now, with this, we come to the end of the insightful discussion. We are truly grateful to all our respective panelists for sharing their knowledge and experience with us today. Thank you very much.
Ladies and gentlemen, let's put our hands together and give a huge round of applause for our distinguished panelists. Thank you so much to our respected session chair and all our eminent panelists for sharing such valuable perspective with us. I would request our distinguished dignitaries to please take their seats down the stage.
No, just wait. So, thank you for giving me the opportunity to just give my concluding remark, whatever we have discussed. We should be thankful to LPU for providing this beautiful session. Yeah, that will do. Because in this session, we scientists, educators, policymakers, regulators, we got the chance to discuss about different aspects when we are using animals. They could get the chance to interact with our young minds, so that how to enhance the ethical responsibility as well as scientific excellence. So all our panelists, they were in agreement that whenever or for any purpose, whatever we are using animals, whether it is small lab animals, fishes, or skid mice, as madam has said, for doping also. So important thing is quality of the animals. Then you need to have trained manpower, which is very important. we can use AI and softwares in management of animal facilities, right? Because there are so many, that way, we can reduce the timeline for manpower also, because everything can be maintained. Regarding ISC approvals also, there are many softwares for management of ISC system as well as animal facilities. Then Dr. Nirmal and Dr. Pawan again, they have stressed that we all have regulatory, ethical and scientific responsibility when we are using any type of model. Like whatever regulatory guidelines are there, we have to follow those guidelines. Scientific means whatever results we are getting, we have to report those, right? We should not worry if we are getting negative results, even then we have to report. So in the end, I will request all the stakeholders, let us continue to collaborate, innovate for better science, scientific excellence, but it should be in science should be, progress in science should be in uniformity with the principles of humanity as well as sustainability. So with these words, I would like to thank again to all the panelists for your critical comments. Thank you very much.
That was indeed a thought-provoking and enlightening discussion. Now, I would like to invite Dr. Bimlesh Kumar, Organizing Secretary and Convener of EESAR, to kindly present the conference report highlighting the key discussions, achievements and outcomes of this remarkable event.
Good evening, everyone. The conference aimed to promote ethical, humane, and sustainable animal research, aligning with the Sustainable Development Goals. It is served as a multidisciplinary platform for researchers, academicians, and professionals to exchange innovative ideas and share advancement in animal welfare and ethical research practices. So in this particular conference, the total number of abstracts which is received, that is the 482, and abstracts selected for the presentation was 172. Abstracts were reviewed and evaluated by a panel of expert reviewers from across the country. Evaluation panel include experts from Maharashtra, Haryana, Madhya Pradesh, Punjab, Jammu and Kashmir. Participants represented a wide spectrum of Indian states including Punjab, Haryana, Himachal Pradesh, Madhya Pradesh, Gujarat, Uttar Pradesh, Jammu and Kashmir, Andhra Pradesh, Telangana, Tripura, Odisha, Maharashtra and West Bengal. And the conference was graced by the presence of several eminent personalities including Chief Patron Dr. Ashok Mittal, Member of Parliament Rajya Sabha, Honorable Chancellor Lovely Professional University, Patron Colonel Dr. Rashmi Mittal, Pro-Chancellor Lovely Professional University, Chief Guest Dr. S.K. Datta, Joint Commissioner Animal Welfare and Member Secretary CCSEA. Guest of Honor was Professor Dr. Atul Nasa, Pro-Vice Chancellor SGT University, Gurugram, Haryana and President IPGA. Eminent guest was Professor Dr. Vijay Bhalla from SGT University. Keynote speakers Dr. Neeraj Khatri from CSIR IMTech, Dr. Vivek Tyagi from CCSEA, Dr. Puneet Kumar Bansal from Central University of Punjab, Patinda. Distinguished guests include Dr. L.D. Singla, Dr. Amritpal Singh, Dr. Dharmendra Ahuja, Dr. Damanpreet Singh, Professor Dr. Alka Beotra, Dr. Harish Chandir, Professor Nirmal Singh and Dr. Pawan Krishan. Evaluation and awards, just now it will be announced, and the organizing committee extend heartfelt gratitude to chief patrons, CCSEA, IPGA and their officials. Sincere appreciation is also extended to all faculty members, staff members, LOC members, student volunteers and participants And my special thanks goes to my core team, Dr. Amrinder Kaur, Vandana Kalsi, Dr. Chaitanya, MVNL Chaitanya, Dr. Dilip Singh Baghel, Dr. Navneet Khurana, Dr. Neha Sharma, Mr. Navneet Duggal, Dr. Rajesh Kumar, Dr. Narendra Kumar Pandey, Dr. Shetu Wadhwa, Dr. Saurabh Singh, Dr. Sachin Kumar Singh, Dr. Palvinder Kaur, Dr. Yana, Mr. Subham, Mr. Rakesh, Dr. Charanjit, and Dr. Vikas Gulati. Lastly, a special thanks to Professor Dr. Monica Gulati, Executive Dean, Registrar, Lovely Professional University. Thank you so much.
Thank you so much, sir, for the conference report.
Now we will move towards valedictory function, and I would like to call Dr. S.K. Datta, Dr. Monica Gulati, Dr. Bimlesh Kumar to deliver memento and awards to our distinguished guests. Please give a huge round of applause to them.
Now, I would like to invite on the stage Dr. L.D. Singla, Guru Angad Dev Veterinary and Animal Sciences University, Ludhiana.
Give a huge round of applause to him.
Please give a huge round of applause for him.
Dr. Amritpal Singh, Guru Nanak Dev University, Amritsar. Then Professor Dr. Alka Biotra, Global Anti-Doping Scientist, former Director, Vada Labs, India and Qatar.
Next we have Dr. Dharmendra Ahuja, Principal Mental Pharmacology, Series Software, Bureau of Health and Education Status Upliftment, Rajasthan. Dr. Harish Chandra, Deputy Director, Quality Control, Natural Institute of Biologicals, Noida.
Please give a huge round of applause to him. Now, Professor Dr. Nirmal Singh, Professor and former Head Department of Pharmaceutical Sciences and Drug Research, Punjabi University, Patiala.
Next we have Professor Dr. Pawan Krishnan, Professor and former Head Department of Pharmaceutical Sciences and Drug Research.
And at last we have Dr. Damanpreet Singh, Principal Scientist, CSIR Institute of Himalayan Bioresource Technology, Palampur.
Can we please have a huge round of applause for him?
Thank you, everyone.
Now moving further towards the next set of events, awards distribution for poster and oral presentation.
And I would like to request Dr. S.K. Datta, Monica Gulati, and Dr. Bimlesh Kumar sir to facilitate the students with the same.
The awards for the best posters goes to, first we have Priyanka Sayal, for the oral presentation, the first award goes to Priyanka Sayal with a unique ID 1159 from Guru Angad Dev Veterinary and Animal Sciences University, Ludhiana, for oral presentation category. Oral presentation goes to Shubham Kumar with the unique ID 1172 from School of Pharmaceutical Sciences, Lovely Professional University. Next best oral presentation goes to Shalini Singh from School of Bioengineering and Biosciences, Lovely Professional University. Please give them a huge round of applause. Next, best oral presentation goes to Gauri Elias Pooja Nayak from School of Pharmaceutical Sciences, Lovely Professional University. And the last best oral presentation award goes to Navneet Kaur with unique ID 1059 from School of Pharmaceutical Sciences, LPU.
Please give a huge round of applause for.
Moving on to the next category of awards for poster presentation. The first category for graduate level, the best poster goes to Pareksha Arora with unique ID 1225. Pareksha Arora. The next best poster goes to Piyush Arora with ID 1128. I request the awardees to kindly proceed towards the stage. Next awardee goes to Mansirat Kaur with ID 1037.
Please give a huge round of applause for her.
The next awardee we have is Harshit Tara with code 1144. Next award we have for Anshuman Tripathi from Lovely Professional University. Anshuman Tripathi.
Can we please have a huge round of applause for him?
The next best poster at graduate level category goes to Ubaid Gulzar from DRD Group of Colleges, Katua, Rajbagh, with the poster code 1127. The last best poster at graduate level goes to Burhanuddin Nagdawala, Indore Institute of Pharmacy, with poster code 1046. Moving on to the next category for post-graduation level, the best poster goes to Ritika Saha with poster code 1106. Next best poster goes to Abhas Sarangi with poster code 1147. The next best poster goes to Priyash Pandya with the poster code 1155. The next poster award goes to Smriti with the poster code 1165. The last poster for postgraduate level goes to Gavin Jaggi with the poster code 1055. Moving on to the next category at PhD level for poster awardees, the best poster goes to Sonam Sarita Bal from Guru Angad Dev Veterinary and Animal Sciences University with the poster code 1190. The next best poster goes to Deepika Bhavsar from Indore Institute of Pharmacy with the poster code 1100. And the last best poster in PhD category goes to Komal Preet Kaur with the poster code 1044. Now, moving on to appreciate the efforts of LOC members, I would first like to call
I would like to call on the stage Dr. Monica Gulati ma'am. Next we have Dr. Bimlesh sir. Next we have Dr. Vikas Gulati sir.
next Dr. Amrinder Kaur.
Can we please have a huge round of applause for her? Next we have Dr. Vandana Kalsi ma'am. Next we have Dr. MVNL Chaitanya sir. Next we have Dr. Dilip Singh Baghel sir.
Now, I would like to call upon stage Dr. Navneet Khurana.
Next we have Dr. Neha Sharma ma'am. Please give a huge round of applause for her.
Now we have Dr. Navneet Duggal.
Next we have Dr. Rajesh Kumar. Dr. Narendra Kumar Pandey.
Next we have Dr. Palvinder Kaur. Now next we have Dr. Yana. Please give a huge round of applause for her.
Then we have Mr. Shubham.
Next we have Dr. Dilip Singh Baghel, sir.
And now we have Miss Sakshi Saxena.
And now I would like to call on the stage Dr. Charanjit Kaur.
Congratulations to all our winners and huge appreciation to every participant who took the effort to present their ideas fearlessly. Every wonderful event must come to an end, but not without expressing our gratitude to those who made it a success. To deliver vote of thanks, I would like to invite Dr. Monica Gulati, Executive Dean and Registrar, Lovely Professional University, to kindly come forward and express our heartfelt appreciation to everyone who contributed to success of this conference.
Good afternoon. Good afternoon. That's like my children. So, I would like to thank each one of you for being, such good children and at times not so good children also when you were making a lot of noise. So, request you to maintain a better decorum. Anyway, thank you so much for maintaining the discipline, maintaining the decorum and, learning and contributing towards the conference. I would like to, first of all, I would like to thank our chief guest, Dr. S.K. Datta. Sir, thank you so much for being with us also and staying all the time. I know we have tired you a lot of things were going on and we've not let you take any rest. But I think when I looked at your schedule that day, you were probably used to it used to more than this kind of work. So, and I would also like to thank all the speakers. My thanks go to Dr. Singla, Dr. Pawan Kishan, Dr. Nirmal Singh, then thank you Dr. Khatri also for being with us. Thank you Dr. Amrit Pal. The people who have left, I have already personally thanked them all. And thank you Dr. Daman, thank you Dr. Ahuja, and thank you Dr. Chandar. I think some of us were pronouncing it wrongly. Yeah, I could sense that. And so, and you know, the person behind the entire effort, so they are all, you are all set for this dance performance, I think. So anyway, my most important thanks are still left for Dr. Tyagi and Dr. Bimlesh, who have been the authors of the story. And then thanks are also due to all the anchors who have done it wonderfully well. Thank you children for doing it so wonderfully well. And thank you to the coordinators of the panel discussion. Where is that girl? The girl who conducted the panel discussion, she also did wonderfully well. So all the anchors, all the coordinators, thank you so much. And then I come to all the people who have contributed, all the heads of the department. Dr. Navneet, Dr. Sachin is not here today. Dr. Shitu, I was looking at her, she probably didn't have her lunch herself, she was roaming around looking at the hospitality. Yeah, thank you, and all the members of the organizing committee. And thank you for, making it a success. Thank you.
Thank you so much, ma'am. Now that we have celebrated the spirit of knowledge and innovation through our competition and awards, it's time to celebrate the spirit of joy, unity, and talent.
कश्मीर की कांगड़ी से लेकर कन्याकुमारी के कोलाटम तक, गुजरात के गरबा से लेकर असम के बिहू तक, जो हमारे देश की रगों में बहती संस्कृति को सलाम करती है। देवी और सज्जनों, लवली प्रोफेशनल यूनिवर्सिटी के इस गौरवशाली मंच पर राष्ट्रीय सम्मेलन EESAR के पावन अवसर पर हम सब मिलकर एक ऐसी यात्रा पर चलने वाले हैं जहां कदम बोलेंगे, संगीत झूमेगा और भारत अपनी आत्मा से गूंज उठेगा। तो आइए जोरदार तालियों के साथ स्वागत करते हैं हमारे नृत्य कलाकारों के लिए।
से सजा हिंदुस्तान, हर नृत्य कहे अपनी पहचान। केसरिया रंग की ज्वाला जलती, शौर्य और जोश की गाथा कहती। सफेद रंग की शुभ निशानी, शांति और भक्ति की कहानी। हरा रंग जब मुस्कुराता है, समृद्धि का गीत सुनाता है। भारत के रंग नृत्य के संग, संस्कृति की यह अनुपम तरंग। हर ताल में बसती है पहचान, हर नृत्य में झलकता हिंदुस्तान, हर नृत्य में झलकता हिंदुस्तान। वंदे भारतम, शतनमन तुझे, शतनमन तुझे, नमन तुझे, नमन तुझे, शतनमन तुझे, शतनमन तुझे। संकल्प लिए जो भी होंगे, उन्हें सिद्धि तक पहुंचाना है। झेनम तकित तक दापिट गरगिरता, तुम दादा पिट गरगिर। झेनम तकित तक दापिट गरगिरता, तुम दादा पिट गरगिर।
Thank you to all our talented performers for that beautiful and energetic performance. It truly added color and joy to our event.
With that, we come to the end of today's conference. It was indeed been a day full of knowledge, creativity, and celebration.
Now, we warmly invite everyone to join us for the high tea. Thank you so much.